National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by 12 research institutes, have 85 research centers, Executive Secretariat, Executive Inspectorate, Center for Data and Information, Center for Technology Services, and Polytechnic Institute of Nuclear Technology Indonesia. Through the 3 directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies.
Good morning, ladies and gentlemen. Thank you to those who have joined the Zoom room and also official YouTube BRIN Indonesia for the second day of ISIBio 2022. I'm Aulia, will be your master of ceremony for ISIBio. Ladies and gentlemen, before we begin the conference today, allow me to read the guidelines of the conference. Presenters and participants are expected to join at least 30 minutes before the conference started. All participants, both presenters and non-presenters, are expected to join as much as possible all sessions in this conference. Please use the virtual background provided by the official committee. Also, please rename your Zoom username according to the following formats. For presenters, you can write down code of abstract or paper _name _institutions. For the example, P4.3 Octaviana _BRIN. Or for non-presenters, you can write down P _name _institutions. As the example, P Octaviana _BRIN. Please mute the microphone, especially during the presentations of the keynote speakers. And in the parallel sessions, presenters should activate the camera during their presentations. Questions from all participants are delivered using the raise hand, Q&A, or chat feature in Zoom meeting. And when Q&A or chat feature selected, please deliver your questions according to this following format: name, institutions, the questions or the name of speakers, and also your questions. Every session will be led by a moderator, and moderator will decide which questions is to be discussed or answered. All participants, presenters, and non-presenters should fill the list of attendees via the link given in each session. Attendees are allowed to know or are not allowed to write down opinion or informations that is offensive, racist, or immortal in nature. An organizing committee have the full right to block or remove any attendee who breaks the above rules. Ladies and gentlemen, we will begin the event. Honorable Dr. Iman Hidayat, Chairman of Research Organizations for Life Sciences and Environment, National Research and Innovation Agency, the Republic of Indonesia. Honorable Head of Research Center for Applied Microbiology BRIN, Head of Research Center for Genetic Engineering BRIN, Head of Research Center under Research Organizations for Life Sciences and Environment BRIN, distinguished speakers and moderators, representatives from ministries, institutions, and universities, and all distinguished guests, ladies and gentlemen. Very good morning from Jakarta. Such a great time for me to welcome back every one of you to the second day of the 9th International Symposium of Innovative Bioproductions Indonesia on Biotechnology and Bioengineering 2022 ISIBio. Ladies and gentlemen, ISIBio has been held annually since 2012, and this year is proudly organized by Research Organizations for Life Sciences and Environment BRIN, bringing the theme of strengthening for bioeconomy through applied biotechnology, bioengineering, and biodiversity. This conference is collaborations with Indonesian Biotechnology Consortium and also ASEAN Federations of Biotechnology Indonesia, and supported by PT Berkaniaga Medica and Nutrilab. As the initial of the agenda for today is keynote speech. Therefore, may I invite Dr. Puspita Lisdianti as our moderator to lead this morning's sessions. But before that, allow me to read Dr. Puspita Lisdianti's profile. Dr. Puspita Lisdianti is a senior researcher at Biosystematic and Evolution BRIN that has interests on biofilm for health and environment, marine biomicrobiology, cacao fermentations, and also functional foods. She obtained her bachelor degree of molecular biology and agricultural chemistry at Tokyo University of Agriculture, Japan, and continued her master and also PhD at the same university, both majoring biosystematics and agricultural chemistry. Throughout her career, she is also awarded Habibie Prize at 2020, Satya Lencana Karya Satya 30 in 2013, and also Satya Lencana Pembangunan in microbiology. Ladies and gentlemen, please welcome Dr. Puspita Lisdianti. Ibu, time is yours to lead the plenary sessions.
Thank you very much, MC Mbak Aulia. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everybody. Honorable Chairman of Research Organization of Life Sciences and Environments, Director of Research Center for Applied Microbiology, Genetic Engineering, Biosystematics and Evaluation BRINs, Chairman of this symposium, and all of the committee. Also, honorable speakers and all participants in this symposium. Ladies and gentlemen, I am Puspita Lisdianti, the moderator of this morning symposium. Our keynote speaker today, the first is Dr. Muhammad Sahlan. He is as a representative of Professor Heri Hermansyah, the President of ASEAN Federation of Biotechnology Indonesian Chapter and Dean of Faculty of Teknik Indonesia University. Let me to read Dr. Sahlan CV briefly. Dr. Muhammad Sahlan is a researcher at the Bioprocess Technology Department of Chemical Engineering Universitas Indonesia. Obtained a doctor and master degree at the University of Agriculture and Technology, Japan in the field of protein engineering. Obtained a bachelor degree at the ITB in the field of chemistry. Awarded first winner of invention in beekeeping field, ASEAN Apiculture in 2018, top 107 Indonesian Innovator Business Innovation Center in 2017, gold medal IGEM In 2014, Roseno Award, Roseno Octroi Bureau, and the National Spice Board in 2013, and Superior Researcher Indo Food Riset Nugraha in 2011. Research interest is waste treatment, waste audit, resource recovery, biofilm, phytoremediation, and PICO remediation. His Scopus H index is 14. Before the presentation start, let me inform you how the presentation will go. First, Dr. Sahlan will be invited to present the material for 30 minutes. After that, there will be a question and answer session for 10 minutes, followed by conclusion. Now, allow me to welcome Dr. Sahlan with the topic Development of Research Collaboration between BRIN and Universities on the topic of biotechnology, bioprocess to improve the bioeconomy sector. Dr. Sahlan, time is yours.
Thank you very much, Dr. Puspita, and also the Master of Ceremony, Ms. Audia. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everybody. I think I have to share my screen for the presentations. Let me to share. Did you see our screen?
Yes, sir. It's already full screen. Thank you very much.
Thank you very much. Okay, recently, now I'm a representative of Professor Heri Hermansyah as the Dean because he have another schedule in this time, and he implied maybe today in this time. So I'm sorry he could not present his presentation. Okay, as Dr. Puspita introduced that in this time, I would like to present about the development of research collaborations and between BRIN and universities on the topic of biotechnology, bioprocess to improve the bioeconomy. In this time, I will focus the presentation or the universities in about the Faculty of Engineering, Universitas Indonesia in collaboration with the BRIN. Recently, I'm also head of the Entrepreneur Innovations Unit and also Reverse Engineering Center that have the correlation between this presentation. Okay, talk about our Faculty of Engineering. We already established on 17 July 1964, and in that time we established 3 departments: civil engineering, mechanical engineering, and electrical engineering. Continue 1 year later, we also established 2 departments: metallurgy and material engineering and architecture. In 1985, we also established the chemical engineering department, started by petrochemical department in that time. And in the 1999, we also established the industrial engineering. Recently, we have a 13 undergraduate program, 2 professional program, 12 master program, and 7 doctoral program. So in detail about the study program in Faculty of Engineering Universitas Indonesia, we have undergraduate program about a certain regular program, a parallel program, and 7 international program. In the professional program, we have 2 professional program. This is for engineer and also architect. And for the master program, we have 12 master program and 7 doctoral program. So this is the detail of the 13 undergraduate study program from the civil engineering and also mechanical engineering. This is the first in study program in Faculty of Engineering. So all the 7 departments, we have the undergraduate and also international undergraduate program. The international undergraduate program, we have the single and double degree. And every department, we have at least another 1 study program. For example, for the chemical engineering, we have also bioprocess engineering study program. That this study program have very related with this topic about the collaboration between BRIN and university in the field of the bioprocess and biotechnology. And also related to the bio, we also have the biomedical engineering. This is for the postgraduate or postgraduate study program. We have 21. For the professional program, we have engineer and architect. For the master program, we have the civil, mechanical, electrical, metallurgy, and also environmental engineering. We also have the master program for this field. And for doctoral program, we have the 7 doctoral program. This is related to the department in Faculty of Engineering. So since this year, our dean, Professor Ali Hermansyah, is the new dean of the Faculty of Engineering, and he know that the condition in the world is very accelerate and the change, and also we have make the strategy how to challenge the accelerated change. So this is the condition in our world. The first is about the volatility of the world, uncertainty, complexity, and also ambiguity of the condition in the world. So based on this condition, we need to collaborate and partnership with other institutions, with other stakeholders. We know that we have the now in disruptive era and also new normal. We have global competition and national competitiveness. We have also industry 4.0 and digitalization, and we need the multidiscipline how to challenge our world in recent condition. So we initiate this challenge by the name of the entrepreneur initiative. We have to challenge. We have the good input in our Faculty of Engineering. We have the students, we have facilities and others input in our Faculty of Engineering. So we have to make the good impact. We have to increase our service excellence, customer satisfaction culture, reputations, glory on the nations, and also revenue for the economic and advancement. So based on these conditions, we decide 3 strategies. The first is empowering engineering entrepreneurship. The second is impactful research and innovations. The third is modernization of engineering education. Based on the 3 strategies, we have 11 excellent priority programs. The first is the organization of multidiscipline engineering, reverse engineering center, engineering seed fund and grant, engineering revenue recognitions, database engineering networks, organization strategic engineering educations. So because we focus on also in educations, we have to increase the quality of the organizations, and also virtual engineering education facility. As you know that the new era need this conditions. And engineering professional program partnership and collaboration package, international academic recognition and engineering student entrepreneur program. So by this 11 excellent priority program, in this time, we focus on the impactful research and innovations by using reverse engineering strategy. So for the impactful research and innovations, we have a platform for the multidiscipline technology development and product co-creation and reverse engineering. This is the new center in Faculty of Engineering Universitas Indonesia. We have to collaborate with the alumni, society, community, and media, government, business, and also industry. So we have the researcher, we have the facilities and also students, we have inventions, fundamental research funding, and also for the local and global market, we have to innovative solutions, and again, revenue income to increase our quality of the output, and asset equity and also license of the intellectual properties. Based on this strategy, we have also the product after we use this strategy in reverse engineering. We learned from this pandemic COVID-19. We have one good example in this case about the ventilator. So by reverse engineering, we have an activity to increase the value of the technology product, either by modifying or by adding features of the technology. For example, in the first 2000s, in the first time, we make the laboratory prototyping one in this stage, and then after about 6 months, we succeed to use to make the prototype 2, and after 1 year, we succeed to production in industry. So this is one of the good example from the laboratorium or from the lab to the industry. We study the original product, and we analyze the product and engineer. We make the prototype and we establish the new product by this scheme of the reverse engineering. By this reverse engineering, we make this center. In this center, we collect the demand from the industry and also from the community. We design the product, and we make the seed fund also. We give the fund for the researcher and also for the team, from the researcher and also from the students that want to make the reverse reverse engineering product, and the output is the intellectual properties. Yeah, we synergy with our another institution is LemTech, with the alumni and partnership industry and sponsor. And with another scheme and the license, we also make the startup and incubate for the technology in implementations. And we hope we can increase the revenue of the market revenue for the Faculty of Engineering. This is another product that we have succeed in the pandemic era. Especially, this is the product for in health, because in these conditions, the product of health is very useful or very good in the market. The first is the Copen 20. Copen 20 ventilator is the first succeed the product, and this product is collaborate not only Faculty of Engineering, but another institution and also the government, how to develop the product and accelerate this product from the laboratory to the industry or to the market. The another product is air purifier with non-thermal called plasma method. This is also the reverse engineering product from Professor Bismo that he expert in the plasma technology and apply this plasma to the air purifier. And this product is also already licensed to the company in Indonesia. Another product is gas portable incubator. It's quite it's a little bit different with another product. These implementations in the society is the scheme is by the community engagement. This product is the incubator to warm premature babies or low weight birth babies in order to prevent hypothermia. And this product is portable design and very simple for mobilization, that very good with the Indonesian situation. And this is another product for the implant, and another product is like propolis, and in traditional traditional medicine. This is also from the chemical engineering. So I would like to talk about the example of collaboration between Faculty of Engineering and BRIN. This case is about the development of dengue paper diagnostic. As you know that the dengue is common in tropical countries, including in Indonesia. As we know that about 390 million of the people is already infected by the dengue, and about 75% of the citizens in the tropical countries have infected by this disease. So in the 2019, it's about 120,000 people infected. It is decreased in 2020, but I think it's because of the COVID-19, so it's very difficult to diagnostic and go to the hospital. So maybe this is the undetected of the disease of the this dengue. So it's maybe the cause by why the number is decreased. So if they treated, the mortality rate is about 2 until 5%. However, if the untreated, it can reach 50%. Yeah, so it's very important to detect in the first stage of the disease. So about 75% of cases are asymptomatic or mild symptoms. Yeah, and symptoms are similar to other febrile illness. And many of them is small diagnosis, and there are 5 serotypes of dengue virus, and the available diagnostic test in our country is real-time PCR. Is difficult to operate in the remote area. So our purpose solutions, we have to make the simple assay is a lateral flow. I think it's already increased also in Indonesia for the diagnostic test. To develop this product, we need 2 proteins. The first is an S1 proteins as a marker or as the target, and as the detector, we need the antibody and also nanobodies. The NS1 is non-structural protein produced by dengue virus when it is entering the human body by this scheme, that after the viral particle enter the cell, the NS1 is produced. So in that time, sometimes the body also can secrete the antibody. for the NS1. So the NS1 can be used as the target protein for the diagnostic, and the high level of NS1 protein were found from the day 1 and 3, making it easier for early detection. For the first, we collaborate with the Dr. Widoretno from BRIN. This is for her dissertation research project, and we study how to make the diagnostic or protocol by using surface plasmon resonance. This is the scheme of the surface plasmon resonance. We can coating or immobilize the antibody, and the NS1 is here, and we can detect if there is the different, but the diffraction, it will be make the signal here. So this is our result that this SPR by immobilize the antibody specific for NS1, we can detect the existing of the NS1, and this is concentration depend. And we compare between SPR with the other method. This is the golden method, the RT-PCR, and using ELISA and literal and SPR, and we found that with the golden PCR is also same result, and we found that the baseline to between the negative and positive result. So because the antibody is also difficult to produce in Indonesia, so we introduced the nanobodies. What is the nanobodies? Are antibodies with the single variable domain located on the happy chain, also shown as PHH antibodies or single domain antibodies. And because this is single antibodies, and without a happy chains, so it's very small, and it's found in camelids and the sharks, and the size also about 10 times smaller compared with the antibody. And so the advantage of the nanobodies is stable in the range of the temperature and extreme pH, and can be expressed in E. coli. So for the first, we developed the recombinant of the nanobodies by transform the sequence of the protein to the E. coli, by using specific plasmid, and we succeed to transform to the E. coli, and also is expressed in the E. coli, and we found we detect by SDS phage and Western blotting that the protein expression is succeed. And also we using the Western blotting, we found that the nanobodies is succeed to produce in the E. coli. And the last, we start to make the rapid detections by using the lateral flow, by using nanobodies as the test line, and this is for the control line, anti-MOS antibody here. And this is the early result that we found that the nanobodies also can use as the antibody for in the dot plot anti-NS1 for the rapid test in the future. And last, I would like for the conclusion that our Faculty of Engineering have to collaborate with the BRIN to achieve the impactful of research, and the collaboration between Faculty of Engineering Universitas Indonesia and BRIN could be also in academic and research sectors. Thank you very much for your attention. We hope we can collaborate, and we can contact our public relation and protocol office in Faculty Administration Center in Depok campus Universitas Indonesia. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Dr. Sahlan, for very informative presentation. Ladies and gentlemen, now are the time for the Q&A session. Please using chat box or raise your hands to give the questions to Dr. Sahlan. Any questions? Okay, if not, I have one question, Pak Sahlan. Bioengineering is must and have to be mastered by biotechnologists, also biologists, microbiologists should master the bioengineering. Because as a biodiversity country, a mega biodiversity country, we have to make a bioproduct by our own self. No other countries would like to teach us, to make a bioproduct. And then, that's a point. And what is the subject? Would you mind to tell us what is the subject in engineering, faculty of engineering and Universitas Indonesia that correlated to the bioengineering?
Okay, thank you very much, Dr. Puspita, for the questions. In the bio-related subject in our the bioprocess and environmental study program and also biomedical study program, we learn about the basics science and also the engineering science. So we learn from the biochemistry, for example, and also we have to learn for the students, learn about the principle of the basic mathematics, physics, and so on. Not only the biology, but the biology is also important, but another is also important related to the engineering basic science. And the second or the third year, we also have the student have to learn about the basic engineering, like mass transfer and separations and so on, and also related to the biotechnology and the bioprocess, like for the cell culture, fermentation, and so on. And in the last year, they also have to take some elections. For example, the election for the protein engineering and for food industry and pharmacy, and also another advanced subject. And in the engineering, we have the two streams. The first is the process streams. So how to process the bioproduct. So this is for the capstones. So the end, we have the capstone course for the product development and process development. So we have two, because in engineering, we have only two, product development and process development. So to increase the knowledge about the economy, we also have the economic engineering also in the time. So maybe this is the subject in Faculty of Engineering related to the bio. For the biomedical engineering is maybe more special because they focuses on biomedical to the medical. So it's more to the medical compared with the general biotechnology or bioprocess. So they have also specific course for the like anatomy and so on. Maybe this is the answer for your question. Thank you.
Thank you, Dr. Sahlan. Yeah, I hope we can we can accelerate our knowledge on bioengineering. Yeah, it's really important from our countries. Any questions or comments? Yeah. Maybe another questions from me, Dr. Salan. Yeah. We are the same universities, actually, in Tokyo University of Agriculture and Technology. Yeah. We BRIN have the nowadays is built good manufacture laboratories, in Cibinong Science Center, if I'm not wrong. Is it can be synergies together with bioprocess engineering in UI, and then we also have some position of a postdoctoral or visiting professor. Yeah, is it possible to work together with the facilities of BRIN?
Okay. Yeah, as I mentioned in our slide, that we can collaborate in academic and also in research. Actually, maybe for the community engagement is also, but maybe I think it's not so much. Yeah, but for the academic and for research is very very good chance for us to collaborate. For example, in the case of the bioprocess, we already have the collaborations, I think, with Dr. Puspita and others. I think BRIN have a good equipment, very good equipment. So why now we also have the collaboration in the case of our research about the rapid test. So our research is in BRIN, in this case in Serpong, in the Serpong. And I think it's also some of the analytical device, is very important. I think it's also, I don't know, for the postdoctoral maybe, postdoctoral. I think it might be our postgraduate students who's already finished their study. They can postdoc or after master maybe, after master. Is it possible for the master, graduate master to not postdoc maybe as a researcher to?
We accept only for the doctoral. Yes,
maybe for the doctoral, because now we already have, even for the doctoral program, is still combined with the chemical engineering, chemical engineering and bioprocess engineering. So the students who want to learn about the chemical engineering in bioprocess stream, so they can also enter in doctoral program in chemical engineering. Maybe I think some of the students after finish can goes to the postdoctoral in BRIN. It is possible, and I think you have the good facilities also for scale up. The scale up is still difficult in Universitas Indonesia, especially for the scale up the productions and for the another assessment like clinical study and so on. So we need the CPOB, for example, or So I think I heard that BRIN have these facilities, and we can collaborate with this. And another one for the undergraduate, because we very strong in undergraduate. For the undergraduate, I think the joint supervision is the good chance, and also for the master course. we can collaborate, and because now is full in our laboratory, so we can have to go to another laboratory. Yeah, thank you.
Yeah, thank you very much, Dr. Sahlan. We got so many information, and then we can synergy together. As a short conclusion, the field of engineering should be learned and should be mastered by biotechnologists, microbiologists, biologists, zoologists, yeah, because you by mastering the bioengineering, we can deliver our product from the lab to industry. Yeah, thank you very much, Dr. Sahlan. Applause to Dr. Sahlan. Thank you very much. Yeah.
Thank you very much, Dr. Puspita. Maybe.
Yeah. Ladies and gentlemen, next we move to plenary session. Today we have two plenary speakers. Our first plenary speaker today is Professor Eni Sudarmonowati. Let me to read her CV briefly. Prof. Eni is a research professor at the Research Center for Genetic Engineering, BRIN. Currently also, Prof. Eni is a visiting professor at the Department of Plant Biology, SEBS, Rutgers University, USA, awarded fund from Fulbright. Also, Prof. Eni is a member of Species Survival Commission, IUCN, Associate Editor, Malaysian Applied Biology Journal. Prof. Eni obtained PhD from School of Biological Science, University of Bath, UK in 1991, and also bachelor degree from Bogor Agriculture University, Indonesia in 1985. Prof. Eni awarded Satya Lancana Wirakarya from the President of Republik Indonesia, Best Inventor, 100 Best Indonesian Women Researchers, Ministry of Women Empowerment and Children Protection, 113 Best Innovation in Indonesia, Ministry of Research and Technology, Ministry of Research and Technology. Her research interest in genetic improvement, genetic conservation, biodiversity assessment, genetic engineering, marker-assisted selection, cryopreservation, omics, and NGS. Professor Eni will be present the material for 20 minutes. After the second plenary speaker, there will be a question and answer session for 20 minutes. Yeah. Now allow me to welcome Prof. Eni Sudarmonowati with the topic of presentation, Genomic Approach for Modern Conservation and Genetic Improvement. Time is yours, Ibu.
Thank you, Dr. Puspita Listianti, as the moderator. Good. I think, can you hear my voice clearly?
Very clearly,
okay. Yeah. So, good morning, good afternoon, good evening, wherever you are. Right now in here is, what's the time now? Is 9:41 p.m. in the US. So I'm very delighted to share my talk. Just let me share screen. I think it's here. Can you see my presentations? Slide presentation? Yes.
Yes, Ibu. Clear.
Yeah. Okay. Yeah. The title is Genomic Approach for Modern Conservation and Genetic Improvement. And the outline of my talk would be, as you see on the screen, I will, as the time given to me, only 20 minutes, I will be discussing a bit quickly, but those that you want to know further details, you can refer to my presentation file, because it's already downloaded in the link that already provided by the committee. So now I move on to the first one, development of genomic-based technology. We've been facing major threat, which is global threats, and then now becoming the world crisis. The main one is habitat degradation, climate change, overexploitation and use of species, invasion of alien species, and pollutions, which made us facing loss of biodiversity. And also a number of animal, plant, and fungi species are now listed as threatened by IUCN Red List, and most known species, like 72%, still need to be assessed. Before move on to the what the genomic can do for the conservation, I would like to share with you the progress of the development of the genomic technology since 1966, and then move on to microsatellites in 1990, and then 2000 SNPs. Everybody now use SNPs, and then for population genomics, it's more on 2010. I will be discussing more on this later on. But in here, you can see detection of inbreeding depression that it's difficult to detect using conventional conservation technology. Just to refresh you, we have 3 sequencing generations. This is just to refresh. The first ones introduced in 1977 with medium read length, still low throughput, and then still high cost base. And then the second one between 2005 and 2010, already high throughput, drop the cost already. And then the third one between 2010 and 2014, it's already long read, and the price, the cost even dropped, becoming more cheaper. But then the development still go on with the establishment of NGS. There are some advantages of NGS. You can read it here, a more lower cost. And then they use a maximum number of SNPs, and also the development for genomic assisted breeding, the GWAS, the Genome-Wide Association, using the utilizing next generation sequencing, and also the GPS genotyping based sequencing. Then develop QTL map, genomic selections, and then the modification, genetic modification using CRISPR-Cas9 after the transgenic era. Just to give you a very brief information on the genomic information already available, already sequenced, and you can find it in the NCBI gene bank. You can see here all different organisms, like the most is Animalia, the green one, the most information available, and then which one is the complete sequence already, the one with the red color. So the choice is yours. I mean, depend on the objective that you targeted. So either you're going to use the whole genome sequence or the whole genome resequencing. So it really depends on the target, but the whole genome resequencing offer you the possibility to screen a neutral and functional polymorphism in high genomic resolution. And further development and moving toward functional genomic, I listed the advantages here, 7 advantages, but there are more. And here, including the wildlife and humans, and also the tolerance and quality traits assessment, and also more powerful technology or mechanism for signaling pathway. And now I would like to move on the genomic roles in conservation of endangered species. And here, why there are some endangered species difficult to be conserved, because there are some obstacles like limited mating pairs, inbreeding, and also inbreeding depressions, so that we need to evaluate which species is vulnerable or less adaptability to the local conditions. So that's why we need to use the suitable technology to address this one. So in this chart, you will see this one is more on the conservation flow chart, but the one with the red one, that actually can be addressed wider when we use genomic as compared to conservation genomics, conservation tools. And the one in green, like genetic drift, inbreeding, hybridizations, can be more precisely estimated by genomic technology. And in here, I just listed several advantages of what genomic can do for conservation, genetic conservation, such as precise genomic monitoring of changes in allelic frequency, and then quantifying the effects of genetic drift and natural selections, hybridizations, and so on. And the advantages of new approaches, not dependent on genomic information already available from the closely related species, so you can do from scratch. So for the conservations, I mentioned earlier that some endangered species are difficult to be conserved. So they declined, the population in the wild declined very rapidly. So then we need genomic approach, genomic-based approach. I mentioned some of them already earlier, but here then because it provides the potential to understand the genetic basis of interaction among species, and also genomic monitoring of changes in allelic frequency to quantify the effects of genetic drift, for example. I listed here up to 20 of the advantages to use genomics for conservation. I don't want to read all of them because of the time, but just to mention, among others, it's actually like the adaptation to local conditions, demographic history, and historical effects, size. Oh, sorry, I listed 2 in here, twice, double. And then the phylogenomics and evolutionary history, population substructure, population connectivity, and so on. And this one, because it provides, this slide shows you, because it provides quite straightforward contributions, such as addressing some problems and then offering possible solutions through genomic approaches, such as minimizing adaptation to captivity, predicting the viability of local populations. And also it offers, with all of our role, of course, how to slow down the bend the curve of biodiversity loss. So in here, this curve, it's actually used by UN, United Nations, on the meeting of CBD. So the bottom, the gray one, the gray line, if you just do business as usual, you do nothing. And then the yellow one, increase conservation efforts, so it will bend a little bit of the curve. But the green one, if you do increase conservation effort, plus more sustainable production and more sustainable consumption, then it will bend the curve of biodiversity loss. Among others, the technology is using the genomic approach because I mentioned earlier that we can prevent genetic pollution and also evaluate its conservation values and so on, and also the migration rate. So what a genomic can do for genetic improvement, because most characters actually regulated by various genes, multiple genes, complex traits, so that it's difficult and take a long time to use this conventional breeding. So using the genomic-based approach, then with the technology I mentioned earlier, the next generation sequencing, among others, and also other technology that combining with others, omics and also gene editing system, then it can fasten the way to get the targeted one. So I now move on to research and applications on conservation. So I show here the genomic-enabled discoveries and application on humans, livestock and wildlife, plants, insects, fungi, bacteria, and viruses. For example, for microbiome in health and disease, for fungi, synthetic biology, and others for plants, for genomic selections, also for livestock, and also the diagnostic for human. This one, sorry, this one not very clear, but in here is the example in wildlife and animal, and also what the aspect also already done, has it already been done, for example, on wolves, monkey, birds, and also some plants also. This is just a summary. And in fish, in Amazon, in Latin America, this one, the symbol fish are Paima gigas. The genomic approach can already offer the fishing management and also genetic management for aquaculture, so that this fish that are almost extinct in the wild then can be increase the population in the wild also. And also it provides low-cost approaches and low-cost infrastructure. For Pacific salmon, this one just to give you several examples, also can give you the migration phenotypes, so that the management, conservation management can be adapted or applied using SNPs. And for Tasmanian devil, in Tasmania, this one almost extinct in the wild, but using genomic approach, the GWAS and also the SNPs, then it can be prevented from extinctions, and now they can be, it's raised the population. And in bird, also in this one, the endemic and also threatened birds in New Zealand. So now using the whole genome resequencing and SNP, so they can produce this one, the conservation, the population also increase in the wild. I mentioned earlier about the adaptability and vulnerability of certain species. So this one, I just give you the case in Japan, that especially in small islands. So this one using the assessment with the whole genome sequence and also the next generation sequencing, then they can provide a recommendation for the government how to preserve this one. And also another plants on rhododendrons, also they provide the government to give effective conservation strategy. Because I'm now in the US at Rutgers University, so I know and also witness the research on this one on conservation, but as well as also for breeding of cranberry. It's famous for sourness, but it's has abundant health promoting compounds. So using a bulk segregation approach with SSR and QTL, and also the GBS, the genotyping based sequencing, so they can produce they can conserve and also produce improved materials of cranberry. For woody species, sorry, not many people working on this because it's a long life cycle, but I just listed the one in Indonesia just to give example, but there are many abroad, internationally, genetic diversity, population genetic assessment and genome sequence of threatened species of Indonesian timber, such as the ironwood, the Aeschylodirexolon swageri, and also the woody species like Mangifera casturi. This is already almost extinct, but now can be propagated, and also the assessment already been done. And transgenesis and the genetic stability test. So this one, the impact of logging and the impact of forest fragmentation already analyzed, assessed using genetic markers in the past, but still the recommendations being used by the government that we need to increase the population in the wild. And this one on Rattan Manau, and also other species under Diplocarp. And now move on to the research and application on genetic improvement. So combining between gamma irradiation and microsatellite markers, two species of Acacia mangium and Paracentus falcaria can be produced, improved one, the mutant one, the straightness of the stem and the timber, and also the permanent branch, and also the height, and also the height and the branch from the surface, the branch height. And to confront with SSR, and then this one using ISSR on cassava to several traits, related to several traits, and also the SNPs for side gene regulating beta carotene also already been done. And also detecting the transgenic plant, this one trees on the less lignin content and high cellulase already been expressed, and this one on at least 4 or 3 species in Indonesia. One of them is endemic Indonesia, the Paracentus falcaria, and the overexpression of the genes stable after 8 years. We tested and also planted. They're still able to regenerate and still stable, confirmed with the molecular analysis. So since again, I'm at Rutgers University, I witnessed, I came with them, this Professor Jim Simon and Dr. Rong Di produced gene-edited basil. Basil is Indonesia like kemangi, resistant to downy mildew disease. I came with them to the experiment station there. So here, you can see the improved basil, very fresh and green. At the back, the one that I put the green light or the green pant or something, that's the wild type. It's already devastated by the downy mildew disease. So it's quite significant, the effect of the gene CRISPR gene edited basil. So this one using the gene homoserine kinase. And for rice, for sweet potato, for wheat, this one from USDA also in US. So they use a different kind of technology like GWAS, and then also other markers for the traits of grain dimension, cooking quality, beta carotene, and others like drought tolerance. So it's really depend on your target And also this one for fruit species, durian, volatile sulfur compounds, and papaya for the fruit weight, fruit length, and so on. Banana for the carotenoid rich, and other fruit for the shape and juiciness. And I listed here also the progress on functional genomics on wheat. So for tolerance and most for drought tolerance, combining transcriptomic, proteomic, and metabolomic using SNP and rapid markers. And now move on the recent and ongoing collaborations between Indonesia and in this one BRIN and Kyoto University and Tohoku University on here you can see the status endangered, critical endangered, and mostly critically endangered and endangered of 4 species using whole next generation sequencing. And also this one is just I just skip this one one of the detail of the species.
2 minutes more.
And and then now using the white durian, this is the first one. We will be the first draft genome sequence of the white durian. So this one, durian cutiensis and durian oxyanus. And then we will generate the PSMC to detect the demography structure population in the past, like 1,000 years back, and also in the future. This one a new technology as compared to PSMC, but also the MSMC, and then this one another new approach using star wave plots. Of course, there are plus and weakness, but this one does not require whole genome sequence as a reference genome. And then we're going to do another 2 white durian for producing draft genome for the first time also in the world, so hopefully it will be soon. And then for networking and collaborations, we need the all participation of all of us, governments, NGO, funding agency. And these are all different type of collaborations, bilateral, regional, global, and others. So we need to make use of that. I just give you the example in the past, a bilateral collaboration with Indonesia, Netherlands, Indonesia, UK, Indonesia, Zurich. So this one on cassava, on stock compositions, and also for extending the shelf life of cassava. And lastly, before conclusions, this one, what the research result for if is it not used by our users. So this one of the users is the government. So we need to give the recommendation for the conservation. So in these 2 books, there are also genomic approaches, the SSR and other markers that have been assessed, used to assess the conservation. And to conclude, lastly, the genomic approach provides more efficient, this one reduce time and low cost for human wellness and nature, not just human wellness, but also nature. So on the first day, yesterday, you all heard from Dr. Lily, the BCG. So this one, bioeconomy, circular economy, and green economy. And the second one, for this technology, we can provide recommendation for national conservation strategy. We can bridging the gap for breeding of elite clones or improved strains or for microorganisms and others. And also we still have a lot of homework to do because certain technology need increased or robust regeneration protocol for many species, and research need to be adopted. So that's our efforts need to be increased for that. So thank you very much for your attention. So I would be very happy to have your questions. Thank you.
Thank you very much, Prof. Annie, for introduce us your work until today. The next of plenary session is Associate Professor Dr. Insinyur Hasimi Abu Hasan Is it already here? Yes.
Assalamualaikum. Can you hear my
Waalaikumsalam warahmatullahi wabarakatuh. Yes, I can hear
voice
clearly.
clear?
Yes, very
Clear.
clear.
Okay.
Yeah.
Okay. Very much. I
Yeah,
try to
Associate Professor Dr. Insinyur Hasimi Abu Hasan is a head researcher at the Research Center for Sustainable Process Technology, University Kebangsaan Malaysia. And then also Associate Professor at the Department of Chemical Process Engineering, UKM, and also Associate Research Fellow, Research Center and Water Analysis, Faculty of Science and Technology, UKM, Managing Committee Member, International Water Association, and then obtained PhD from University Kebangsaan Malaysia. His honors and awards obtained was Anugerah Bintara Ahli Akademik Harapan, UKM, Most Valued Reviewer for Bioresource Technology Elsevier, Anugerah Bintara Ahli Akademik Harapan, also from UKM. The research interest is on a waste treatment, waste audit, resource recovery, biofilm, phytoremediation, and remediation. Professor Hasimi Abu Hasan, time is yours. You will make your presentation by yourself?
yes, sure.
Okay.
Thank you, Mr.
Thank you very much.
Jefferson. Okay. I will share my screen. You can see my screen, right?
Yes,
So
we can see yours.
just a moment. Okay. 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 Thank you for inviting me to give a speech in this conference. Okay. As presented by the Mr. Chairperson, my name is Hasimi Abu Hasan from Department of Chemical and Process Engineering, and acting as head for the Research Center Sustainable Process Technology. Okay, so my topic today is on the how to treat the river or how to treat the surface water through the biological approach. Okay, just a moment. I try to remove this one. How to? Okay, never mind. Okay. So so this is my outline for the presentation for the speech. Okay, I will start with some introduction on the environment, especially on the river. Okay, and then what kind of the river pollution in the in especially in Malaysian country. Okay, and then the treatment technologies we use to treat the river. Okay, to produce clean water, and then the challenge. Okay, the challenge to use the biological approach. Okay, and the last one, of course, we have conclusion. Okay, as introduction, as a human, we need a water. Okay, because water has an important role in our metabolic process. Okay, so this water it will break down a large substance into a small substance. Okay, so what happens if the water contaminated by the pollutant? Okay, of course it will affect to our body. Okay, So the contaminated water will become hazardous, okay, not only to human as well as to plant, hazardous to the animal, and this kind of pollutant will expose through the drinking water. Okay. So there are a lot of pollution sources in the river. Okay. For example, from the industrial discharge, okay, the pollution from the illegal garbage. This is the common one. Pollution from the illegal garbage. Okay. And then pollution from the sewage effluent discharged to the river without proper treatment. And then from the leachate, okay, leachate landfill, and of course from the agriculture industry. Okay, so agriculture industry can pollute the river through the water runoff, okay, during the watering or during the raining season. Okay. Okay, this is some of the river water pollution. Okay, in terms of chemical compound. Okay, there are 4 categorized here. Okay, the first one is heavy metal. Okay, there are many heavy metal contamination in the river. Okay, for example, plumber, cadmium, or zinc. And then other parameter is inorganic, non-metallic. Okay, so inorganic metallic, the source for this kind of pollution is from the sewage industry, from the industrial industry. Okay, and for example, of the inorganic non-metallic pollutants, nitrogen compounds, sulfate, or chloride. Okay, and then what else? The river water pollution. Okay, endocrine disrupting chemical, EDC. Okay, and disinfection byproduct. Okay, these 2 categories are under micropollutant types. Okay, we could not see by our naked eye. We can analyze both categories using high-end equipment and not very easy to analyze. So this is the current issue in the river, okay, around the world. Okay. So and then other kind of the water pollution are physical. Okay. So the physical contamination contribute by the garbage. Okay. And then once we throw the garbage into the river, so it will contaminate the river, of course, as well as the color. It will create the bad color, the black color, the brown color of the river. And then we can see the turbidity, the clearness of the river become reduced, okay, through the turbidity measurement. Okay, this is some example, the water pollution in Malaysia. Okay, some news, okay, about the contamination. Okay, and that news in Malay, I think you can understand. Okay, you can see the fish in the river, okay, died because of the pollutant. The river water become toxic. Okay, and not only die, some aquatic plants also become yellowish, start to decay. Okay, and some other example. Okay, in Malaysia, there are many. Each year we can hear about the water contamination, not only in Malaysia, also in other countries, especially in developing countries. Okay, there is some other example. So you can see this in English. The river water turn green. Okay, this is because of the dye discharge. Okay, from the textile industry. So they discharge the dye directly to the river without any proper treatment. Okay, so in Malaysia we have a conventional treatment. Okay, to treat the river water. if the river are very contaminated at high level, of course, the conventional treatment could not be operated for the production of clean water. Okay, so in Malaysia we have the treatment of the grid chamber. Okay, this one, and bench drain. This is to remove the garbage. Okay, or to remove the large particle. Okay, for example, the dead leaf, the trunk of the plant. Okay, and then we have the aerator here. Okay, aerator is purposely to reduce the light heavy metal. For example, the ferrum, the manganese. Okay, and then followed by the flocculation and sedimentation tank. Okay, this is to remove the dissolved organic material. Okay, for example, to reduce the suspended solid, to reduce the dissolved metal, so we use the flocculation process. And then we have a sand filter, okay, before the end of the treatment. Okay, sand filter to remove residual suspended solid. And the last process, we treat the water by the chlorination. This is to ensure there is no microorganism contamination in the water. Okay, so our purpose, okay, why we develop a technology to treat the river water using the biological approach is in the current technology we could not remove the nitrogenous contamination. For example, we could not remove the current technology could not remove the ammonia, could not remove the nitrate, could not remove the nitrate. So by having the biological approach, okay, so those pollutants could be removed effectively, and at the same time the water treatment in Malaysia continuously operated, okay, without any shutdown. So this is what we propose and what we study in Malaysia in my department. Okay, biological treatment is one of the advanced treatment. Okay, so it started popular since 1900, but not for the water treatment. It's popular for the wastewater treatment. Okay, so the application is still limited around the world. Okay, but for the developed country such as US, Canada, they have very well knowledge on the technology for the drinking water production or for the river treatment. Okay, so of course, when we're talking about the biological approach, we have to make sure that the process use non-pathogenic bacteria. Okay, so this kind of bacteria will act as biocatalyst for the biochemical oxidation, and then it can degrade the pollutant in the river. So the technology could be proposed before the chlorination process. Okay, because if you propose at the end of the process, it might be contaminated with the microorganism. But if you propose before the chlorination process, so the microorganism contamination could be avoided. Okay, so now we go to the what kind of the technology for the biological approach. Okay, so we can use the slow and rapid sand filtration. Okay, so we have published a few articles on this technology, and this technology could remove the light heavy metal. For example, it could remove the manganese, it could remove the ferrum, it also could remove the plumbum, okay, in the river water. Okay, so but the lacking of this technology, it could not be operated if the heavy metal too high in the river. Okay, too high means it could not exceed 2 ppm. Okay, if the concentration exceeds 2 ppm, so we could not use this technology. Okay, because the heavy metal could provide a toxicity to the microorganism. Okay, so this is some explanation about the sand filtration. Okay, there are two types of sand filtration. The first one we call slow sand filtration, and second one is rapid sand filtration. Okay, so this process used normally we used to filter out the suspended solid and light heavy metal. Okay, so the sand filter, okay, that fail in the reactor, okay, will act as a support for the biofilm growth. Okay, once the biofilm grows, it can provide a space for the biological activity. Okay, and we can design the sand filtration in a column shape or in a rectangular shape. Okay, this is the biofilm layer on the sand surface. This is in German language. Okay, we call it smart Dutch. Okay, and this biofilm layer contain extra polymeric substance. Okay, so this substance could react with the pollutant through the bioaccumulation, through the bioabsorption, and of course we can produce the clean water. And this is some of the performance for the biosand filter to clean the river. Okay, especially the ferro and manganese as a light heavy metal, as well as the ammonia. Okay, we can also treat the ammonia if the ammonia level high, more than 3 ppm, so we can use the sand biofilter. Okay, next is biological activated carbon. Okay,
you have 5 minutes more, Sensei. Yeah.
Okay, I will speed up my presentation. Next is biological activated carbon. So this kind of technology use the activated carbon to absorb the pollutant. Okay, so there are two mechanisms for the biological activated carbon. The first one is aerobic process, and the second one anaerobic process. So the activated carbon or the adsorbent could be regenerated through the microorganism role. Okay, so the microorganism will consume the pollutant, and at the same time the pollutant from the activated carbon will consumed back by the microorganism for their growth. Okay, this is the performance. It can reduce the micropollutant. Okay, something like EDC. Okay, and then trickling filter, another treatment. Okay, this is not a common technology for the trickling filter, but we can use the trickling filter to remove the manganese, okay, iron, and also ammonia. Of course, this technology will involve the microorganism, something like manganese oxidizing bacteria, ammonia oxidizing bacteria, for example, Nitrosomonas, Nitrobacter. Okay, and also we can use the biological aerated filter. We have published more article for this kind of technology. Okay, this will involve the using of floating carrier. Okay, the floating carrier will act as a house for the bacteria to grow, to attach, and at the same time it can provide a large surface area for the biofilm, for the contaminant removal. Okay, and this is the performance. And then we also could use membrane bioreactor. This technology combination of the bacteria, especially bacteria, and the membrane. Okay, and there are two type membrane technology. Okay, we call it as attached growth membrane bioreactor, and we can also set as ion exchange membrane bioreactor. Okay, so normally we use the membrane bioreactor at the end of the process before the chlorination. Okay, And also we can use the moving bed by film reactor. Okay, this technology combining the activated sludge process and also with the aeration, we provide some aeration for the bacteria. Okay, and the aeration could create the dissolved oxygen concentration in the contaminated water, okay, normally at 2 until 5 ppm. Okay, so there are also some integrated biological approach. Okay, with other technology. Okay, for example, we integrate the membrane. Okay, membrane bioreactor with the coagulation process. We add some chemical before the treatment. Okay, so the chemical will reduce the suspended solid or will reduce the light heavy metal, and the treated water will go to the membrane and it can increase more the pollutant removal. Okay, and then we also integrate with the adsorption technique. Okay, between the adsorption and the membrane. Okay, we call it as biological activated carbon membrane bioreactor. Okay, so this is the combination. And then what is the challenge in using biological for the drinking water treatment? So we have to face, we have to solve how to make sure the treated water are free from the pathogenic microorganism contamination. Okay, this is the challenge. We should have a frequent monitoring on this parameter. And then the selectivity of the microorganism towards the pollutants. Okay, not all microorganisms have capability to remove the pollutant. So we should provide the system with a good microorganism that could remove what parameter we need. Okay, and then the third challenge is to maintain the growth of the microorganism in the system. Okay, this is very challenging, and of course, once we produce the drinking water or we treat the river, what is the consumer perspective? Okay, are they worried about the contamination of the microorganism or not? Okay, so we have to do some survey for this. Okay, and at the end, for the conclusion, of course, this is a new technology or our future technology for the river water contamination, as well as especially for the developing country. And there are many area that we can explore, and of course, this technology could be growth well in developing country. Okay, thank you. I think that's all my presentation. Before I end, I want to say something. The earth is beautiful. Let us maintain the beauty by caring for the environment. Okay. Okay. Thank you very much, Mr. Chairperson.
Thank you very much, Dr. Hasime Abu Hassan. Ladies and gentlemen, now are the time of Q&A session. Please using chat box, chat feature of the Zoom and raise your hands, and then from whom the questions will be delivered. We have 10 minutes for the question and answer session. And then there is a one question from the chat book to Prof. Annie. According to your experience, what method is the most effective and efficient in transferring the desirable gene into the plant? From Ibu Elok, BRIN.
Okay, thank you for the questions, Ibu Elok. Yeah, so nowadays, because of the development of CRISPR-Cas9, this is the most effective way. I mean, efficient, because efficient when we talk about the resources. So it's quicker in terms of time. I mentioned in my slides, I showed to you all the successful research using CRISPR-Cas9 on basal at Rutgers University in the US. So this one, they only needed 2 years to get the gene-edited plants as compared to the one that using the conventional breeder, they need 7 years. And then how to deliver the gene after it's cut, by CRISPR-Cas9. For my experience also, and also when I'm here at Rutgers, using particle gun as compared to agrobacterium tumefaciens, because when you use agrobacterium, it's almost most of the time you will be like deleting or to get rid of the bacterium from the plants. I mean, if the concentration is too high, then your time is consumed for transferring or get rid of the agrobacterium. But using particle gun bombardment is better, because then you don't have to be disturbed with the agrobacterium. So in here at Rutgers University also, they use the particle gun bombardment of BioRad. I mean, last time I used not BioRad because we used the cheaper one, but less effective. So I think we need to have the BioRad also. And also, although either you use the conventional genetic engineering or using the transgenesis or CRISPR-Cas9, again, as I mentioned in my conclusions, one of the list that the regeneration procedures is need to be established. So no matter how high or how advanced is your technology, if you want to improve genetically, you have to establish the regeneration system. Yeah, so that's my answer. I hope that's satisfy you. Thanks.
Okay. Thank you very much, Prof. Annie. And then from the chat book to Prof. Hasimi from Ibu Penny Lestari. The result of this research are very good and protecting the earth from contaminants. Is the use of bacteria of microbes to treat pollutant also safe for human? Or how to manage these microbes? How far have the results of this research progressed? Has it been applied in product form? How to get it? Please, Dr. Hasimi. Yeah.
Okay, thank you for the question. Okay, apologize. Okay, there are few questions, right? Okay, how to manage this microbe? Okay, during the treatment, okay, we could monitor the growth of the microbe, okay, through the what we call it mixed liquor volatile suspended solid, okay. Or we can measure through the adenine triphosphate analysis, okay, ATP. So this value represent the growth of the microorganism. Okay, once we produce the treated water, okay, after the biological treatment, we will treat with the chlorination. Okay, so the chlorination will destroy all the microorganism, and we will make sure after the chlorination process, there is no microbe contamination in the water. Okay, this is what we have done. Okay, and then so of course the safety of the treated water to the human. Okay, so this is the challenge that we have faced. Okay, so the consumer will worried to consume this water because they think it contaminated with the microorganism. Okay, so we have to convince the consumer or the human that the water is safe. Okay, because we are using the non-pathogenic bacteria. Okay, not pathogenic bacteria. Okay, so and we also control with the chlorination. Okay, before we distribute the water to the human. So through this control, okay, so we can ensure that the water are safe to the human. Okay, so and then so far this project is still under research progress. Okay, not apply in a real scale or pilot scale. Okay, of course, there are more study to be explored. Okay, especially the consumer perspective. Okay, and there are one question about the economic. Okay, so for the economic test or analysis, this still ongoing. Okay, but at the lab scale, not at the pilot or real scale. Okay. Okay.
Yeah.
That's my answer.
Okay, and I want more answer. How about the study from economic side, when it will be applied?
Yeah, of course, in terms of economic side, okay, we use less chemical, okay, and then it easy to operate, okay, easy to operate. That doesn't mean we do not need a competent staff or competent operator to operate. Okay, still need the competent operator, but easy to operate, easy to learn. Okay, just to competent how to manage the microorganism growth in the system. And of course, economic side is more cheaper compared to other technology. Okay.
Okay, thank you very much, Dr. Hasimi. We will be finished for this plenary session. Thank you very much, Prof.
Yes,
Any.
there are two. Sorry, there are two.
Oh yeah.
There are two raising hand.
Oh, I'm sorry. Oh yes.
Yeah.
Pak Panji and Ibu Yeni, please. First, Pak Panji.
Do I still have time, by the way?
Yes, we still have 1 and 2 minutes.
All right.
Okay.
All right. I'm just going to make it quick. I have one question for Bu Eni and one question for Pak Hasimi. For Bu Eni, I am studying microbiome now, and I learned that the loss of the microbiome would influence the loss of the high organisms as well. Do you think that targeting the microbiome in addition to the genetic identity of the microorganism would help to preserve the endangered species? And for Pak Hasimi, I am now doing a project with KU Leuven about the use of microbial inoculant and as well as the horizontal gene transfer in microbiome to reduce the pollutant. And one of the key challenges that we face is to maintain the survivability of the microbial inoculant. How did you maintain yours in your system? Thank you very much.
Thank you very much. Please give
Can
the
I
short answer?
Yeah, very short one. Okay, yeah, before I also short, because of the time. Yes, definitely studying the microbiome will affect the degrading or increasing the populations or enhance the loss of biodiversity or maybe improve the population size. That's why the study in microbiome is very important, and also because it's also related to the gut, even in the intestine of the animal or wildlife or even human. So that need to be studied further. Yeah, so that's why the study in microbiome is very important, because it's related to the extinctions or the survival of the species. Yeah. Yeah, that's my quick
Thank you,
answer.
Ibu Eni. Dr. Hasimi, please.
Okay. Okay. How to maintain the growth of the microbe in the reactor? Okay. What we do is, the first step is we have to make sure the incoming river water to the system is not at very high contamination. Okay. So we should remember this technology is used not at the first stage. We could use at the second stage or the third stage. Okay. If the concentration of the contaminant is very high, so it will kill or will become toxic to the microorganism. Okay. The contamination depends on what parameter we want to treat. Okay. And for example, if we want to treat the ammonia, okay, we have to make sure the ammonia level after the first stage of treatment, it should be not more than 3 ppm. Okay, so we are talking about the water, so the contamination should be not high, and from that it will maintain the growth of the microorganism, and we can monitor the growth through the mixed liquor, volatile, suspended solid. Okay, that is what we have done in our research.
So by that you mean you get the microbiome to adapt to the environmental stressor that would...
Yes.
Okay.
Yes,
right.
we have to do that. Okay, to make sure it can adapt with the environment. It can have a good removal stability performance.
All right. Okay. Thank you very much, both.
Okay.
Thank you very much. I'm sorry, Ibu Yeni, the time is up. And then thank you so much again, Prof. Eni and then Dr. Hasimi, for the very informative and then inspiring presentation. Yeah, I move this session to MC.
Thank you.
Yeah, thank you so much.
Thank you.
Thank you, Prof. Eni. Thank you, Dr. Hasimi, and also thank you, Dr. Pusvitalis Dianti, as the moderator of the sessions. Ladies and gentlemen, following to the next agenda, we will have the parallel sessions consisting of 5 rooms with different topics, and also each of room will be led by moderators. So the first room will be bioresources and biodiversity. The second room will be nutraceuticals and natural products. The room 3 will be biotechnology and bioinformatics. Room 4 will be medical sciences, and the last room, biorefineries and circular economy. So please kindly choose the room based on your interest by clicking the breakout room feature at your Zoom. If you haven't found it yet, you can click more with 3 dots at the corner of the left of your Zoom meeting, then join breakout room there. The room 1 until 5 are available there. Once again, please kindly choose the breakout room by clicking feature more and then join breakout room. Allow me to read once again the room. Room 1 will be bioresources and biodiversity. Room 2, nutraceuticals and natural products. Room 3, biotechnology and bioinformatics. Room 4, medical sciences, and room 5, biorefineries and circular economy. As you can see on your screen right now, enjoy the sessions.
The voice is not clear. I'm sorry, Ibu Wening, but your voice is not clear.
Now, my voice is already clear?
Yes, you can start now.
Oh, okay. Okay. Thank you. Thank you, Mbak Senli. Thank you for the time and this valuable chance. Hello, everyone, the speakers and also the participants. Assalamualaikum warahmatullahi wabarakatuh. Good morning. I am Wening Enggarini from Research Center for Genetic Engineering, National Research and Innovation Agency. I am the moderator for this session. Now I will call all the speakers one by one. For the first, Mr. Haryo Bimo.
Yes, I'm in here.
Okay. Next, Mr. Baso Didik Hikmawan. Next, Mr. Baso Didik Hikmawan. Oh, not yet. Okay. Mr. Hairul Anam. Already there?
Yes.
Okay. Next to Mr. Rian Haryo Setiawan.
Yeah, hello.
Okay. And the last is Miss Marlin Megalestin Raunsai.
Yeah.
Okay.
Sunny.
Okay, thank you. Maybe Mr. Baso Didik will come later, okay? Okay, I see all speakers already joined. Please note some points for the speakers and also the participants. One, the discussion will be held after all video presentations finish. And two, we are very welcome any suggestion and question. You can type in the chat room or use the raise hand icon. And three, I am also mandated as a judge for the nomination of best paper and speakers. So please all speakers have to give the best shot in this session to get the highest score, okay? Okay, let's start this session. We go to the first speaker is Mr. Haryo Bimo Setiarto. Will give presentation entitled Effect of Types and Concentrations of Stabilizers for Organoleptic and Nutritional Quality of Functional Food Product Velva Champolai Pouteria campesiana. Please play the video.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Dear ladies and gentlemen, my name is Haryo Bimo Setiarto from the Research Center for Applied Microbiology, National Research and Innovation Agency BRIN. I will present my conference proceeding paper for ISIBio 2022 with my paper is entitled of Effect of Types and Concentration of Stabilizers for Organoleptic and Nutritional Quality of Functional Food Product Velva Champolai Pouteria campesiana. I will start from the introduction for this manuscript paper. Champolai fruit Pouteria campesiana is a plant originating from Mexico that can grow at tropical and subtropical climates. The use of champolai fruit is currently still limited, which is only used as syrup, jam, flour, or consumed directly. The taste is sweet like sweet potato, and the intense aroma makes people not like to eat champolai fruit directly. Therefore, it is necessary to diversification of food preparation from champolai fruit to utilize champolai fruit and extend its shelf life. One alternative to using champolai fruit is making velva champolai. Velva is a type of frozen food similar to ice cream but has a low fat content because it does not use milk fat and has a high vitamin C and fiber content. The role of the stabilizer in velva is to bind the water in the dough to form fine ice crystals. Stabilizers function to maintain the body and texture of the product during storage. This study aims to determine the type and concentration of the best stabilizers in the production of Velva Champollion based on consumer preference and to analyze the nutritional value and antioxidant activity of selected products of Velva Champollion. Next, we move to material and methods to produce Velva Champollion. The materials to produce Velva Champollion which used in this study include overripe Champollion fruit, which is yellow in color and soft in texture. The other material is stabilizers such as carboxymethylcellulose, CMC, carrageenan, gelatin, sucrose, and lime. The research design for the experimentally design used in this study was a two-factor, completely randomized design. The first factor is the type of stabilizer with 3 treatment levels, CMC, carrageenan, and gelatin. The second factor is the concentration of stabilizer with 3 treatment levels, 0.25%, 0.50%, and 0.75%. Each experiment was replicated twice, so 18 experimental units were obtained. How to produce Champollion fruit pure? Champollion fruit used in making Champollion fruit pure is the fruit that is overripe and not rotten. Champollion fruit is washed, peeled, and then spread between flesh and seeds. Then the fruit's flesh is blanched by steaming at a temperature of 80 degrees Celsius for 3 minutes. Furthermore, the flesh of the Champollion fruit is added with water and crushed with a blender until smooth. Then Champollion fruit pure was added with 15% sucrose, 5% lime juice, and stabilizer treatment. Then mixed with a mixer for 15 minutes. Then cooked at 5 until 6 degrees Celsius for 45 minutes. After that, it was packaged and frozen at minus 18 degrees Celsius for 24 hours. Then we analyze the Velva Champollion products such as organoleptic test and hedonic test for Velva Champollion product, melting power for Velva Champollion product, proximate analysis for Velva Champollion selected products, analysis of vitamin C of Velva Champollion products, and analysis of antioxidant activity of Velva Champollion products. Next, we move of the results and discussion. The table 1 is show about the sensory quality and hedonic test of Velva Champollion products. Based on table 1, we know that the type of stabilizer has a significant effect and the concentration of the stabilizer has no significant impact on the overall level of preference for Velva Champollion. This means that the type of stabilizer can affect the level of preference of the panelists on Velva Champollion as a whole. The primary purpose of stabilizer in frozen desserts products is to produce a good smoothness and texture, inhibit or reduce the formation of ice cream crystals during storage, and produce a uniform texture. The panelists' level of preference for Velva Champollion overall ranked from 6.03 until 6.87, somewhat like. The most preferred Velva Champollion is Velva with added
Excuse me, Mbak Senli, the video is stopped.
compared to the other treatments has the right combination of sweet and sour taste. Table 2 from results of discussion. Table 2 shows that the nutritional value and antioxidant activity of selected product of Velva Champollion. The moisture content in the selected felva champollion is 75.73%. The high moisture content comes from adding water to produce pure champollion and other ingredients used when making felva champollion. Moisture content in the product is an essential component that can affect the texture, taste, and appearance, and the water content is related to the product's shelf life. Ash content is a mineral element as a residue left after the material is burned until it is free of carbon. The ash content contained in the selected felva champollion is 1.14%. The protein content contained in the selected champollion felva is 1.71%. The fat content in the selected champollion felva is 0.14%. The carbohydrate content in the selected felva champollion was 21.28%. The crude fiber content in the selected champollion felva is 0.11%. The energy total value contained in the selected champollion felva is 93.22 kilocalorie each 100 gram felva. The decreases in energy value can be caused because the champollion fruit used is overripe, so the nutritional value in 100 gram of champollion fruit has decreased. The level of vitamin C content in the selected felva champollion was 21.43 milligram each 100 gram. Decreased level of vitamin C in felva champollion can occur during the process of making felva. Vitamin C is a perishable vitamin during processing, such as blanching, washing, cutting, and blending. The antioxidant activity IC50 of selected felva champollion was 542.59 ppm. Felva champollion is classified as having very weak antioxidant power. Then we move of the conclusion from this conference proceeding. Champollion fruit can be used as material for felva champollion with overall preference ranging from 6.03 to 6.87, somewhat like. The stabilizer was not significantly affect the quality of the texture and the level of preference of the panelists on the texture test overall, but it significantly affect the melting power of felva champollion. The concentration of stabilizer was not significantly affect the quality of texture, but it significant affect to melting power of felva champollion. Felva champollion product selected based on the sensory quality test, hedonic test, but based on melting power was felva with a CMC stabilizer of 0.75%. The selected champollion felva has 75.73% moisture content, 1.14% ash, 1.71% protein, 0.14% fat, 21.28% carbohydrates, 0.11% crude fiber, energy value 93.22 kilocalorie each 100 gram, vitamin C 21.43 milligram each 100 gram, and antioxidant activity IC50 is 542.59 ppm. Thank you very much.
Okay, thank you. For the next speaker is Mr. Basodi
My name is
Mawan. Yes, and he will give presentation entitled Determining Optimum Harvest Time of Coristis species for its maximum antioxidant capacity and pigments production. Please play the video.
Didik Hikmawan, I would like to say thank you so much to you all who have given me chance and times. So today I could present the result of our study about determining optimum harvest time of Coristis sp for its maximum antioxidant capacity and pigment production. This is the background of our research. Natural antioxidants have become scientifically interesting for researchers due to its beneficial health effect. Well, natural antioxidants are used in pharmaceuticals, cosmetics, and also food industries. Therefore, the demand for natural antioxidant is rising. Most commercial antioxidant products are derived from plants. Unfortunately, destructive harvesting typically leads to the resource exhaustion and potentially species extinction for medicinal plants. Therefore, the cosmetic and pharmaceuticals, also food industries, should consider alternative sources of antioxidants. Macroalgae are seen as one of a potential and valuable source of antioxidant. It is because microalgae can produce antioxidant compounds. Also, it is easy to be cultivated and it has rapid generation times, also provide an environmentally sustainable approach to industrial scale production. The antioxidant capacity of microalgae, of course, depend on the concentrations of secondary metabolites such as carotenoid, phenolic compounds, and pigments. Harvesting macroalgae in exponential or in stationary phase of its growth is favorable since these two phases usually results the high cell biomass, and the biomass taken from these phases is used to examine the antioxidant. Therefore, the result mostly reflect a particular growth phase without considering the variations in the antioxidant compounds compositions among phases. So in this study, we use Corycistes sp as one of potential microalgae, and this study set out to identify the precise Corycistes sp growth phase at which the harvested microalgal biomass concurrently has the highest antioxidant capacity and pigment concentration. Well, Corycistes sp was isolated from freshwater lake in Bengkulu province, Indonesia, and cultivated with continuous aeration, illuminated with fixed light intensity, and cultivated in modified AF6 medium. For determinations of antioxidant capacity, we use ABTS inhibition assay, where ABTS radical cation was mixed with Corycistes sp extract and measured it in spectrophotometer at 734 nanometer from 0 to 6 minutes. After get the absorbance of each minute, we subjected the absorbance to this equation, so we got the antioxidant capacity. Also, to determine its growth phase, which later used to observe the optimum harvest time by examining the antioxidant and pigment content from each phase, we plotting the time against the cell number of the Corycistes sp. We make a growth curve by using high-mositometer and also microscope. We use methanol to extract the pigment and measure it by using spectrophotometer for measurement of the chlorophyll A, B, C, D, and also carotenoid, as well as the total chlorophyll. And the antioxidant extractions, we use a hot water extraction technique, which is dried biomass suspended in aqueous, 1 in 5, one dried biomass in 5 aqueous, and after that we use autoclave to extract it. This is the result of our study. As you can see in the screen, that we draw the growth curve of Corycistes sp by plotting the time, days, and the cell numbers. There were 4 growth phases of Corycistes sp, as it could be seen. The first one is lag phase, which is from day 0 to 3. After that, Corycistes sp enter the exponential phase, which is day 4 to 5, and after which enter stationary phase. The stationary phase here, it's from day 6 to day 10 of the cultivations, and after that it entered the death phase up to the day 16. So we use this pattern to examine antioxidant capacity, also the pigment content, from each growth phase. So this is the result for the pigment measurement levels in each growth phase. As you can see, this is the lag phase, log phase, stationary phase, and the death phase. The results show that the total chlorophyll peaked at stationary phase, which is 8.81 milligram per liters, and carotenoids also peaked at stationary phase as well, with the yield of carotenoid was 4.09 milligram per liters. However, in the total chlorophyll, as you can see in the log phase, it is also 8.09, where statistically the level of total chlorophyll in the log phase and stationary phase is not different. Next, in the antioxidant capacity measurement, we found that the log phase showed the highest inhibitions of the ABTS, where in the 6 minutes it could inhibit 99.75% of the radical cation. And this is the conclusions of our study, that the optimum harvest time for microalgae Corycistes sp in order to obtain the highest pigment and antioxidant concentration was when the microalgae reached the final logarithmic or exponential growth phase, which is day 5 of the cultivation. And at the particular growth phase, the antioxidant extract of Corycistes sp was able to neutralize 91.51 to 99.75% of radical cation ABTS. So the pigment concentrations, especially chlorophyll, was also in highest concentration at the final logarithmic phase. However, the carotenoid content was highest when the microalgae grew until the final stationary phase, or in day 10 of the cultivation. Therefore, if it was carotenoid that was expected to be high in the harvested biomass, then microalgae, this Corycistes sp should be harvested when it was in the final stationary phase or in day 10. Well, this is the reference that we use. And I say thank you very much for your attention.
Okay, thank you. For the next speaker is Mr. Hairul Anam. Will present entitled Quality of Spirulina maxima in Different Water Source for Media Cultivation and the Biomass Storage Condition. Please play the video.
Good afternoon, everyone. My name is Hairul Anam from Research Center for Applied Microbiology. Today, I would like to present my research entitled Quality of Artospira maxima in Different Water Source for Cultivation, Media, and Biomass Storage Condition. Artospira, also known as spirulina, is a type of microalgae belonging to the cyanobacter group. It is widely used as a source of protein, vitamins, and pigments. This microalgae known has a high content of protein. Phycocyanin, one type of protein compound included in Artospira, has an activity for antioxidants, anti-cancer, anti-inflammatory, antibacterial, and immunomodulation. Artospira platensis is one of microalgae that widely developed for its growth condition, biomass production, and biochemical composition. Meanwhile, Artospira maxima information is still limited. Because of that, in this study, Artospira maxima was used to investigate its biochemical properties. Although Artospira maxima is known to have antioxidant activity, the content of the biochemical properties produced by the microalgae also depends on microalgae conditions. The conditions that can affect the biochemical properties are the type of growth medium and biomass storage. Because of that, in this study, we investigated the effect of seawater and freshwater to Artospira maxima biomass production and the storage condition. Therefore, this study aimed to analyze the effect of xeric media in freshwater and seawater for microalgae cultivation and the storage condition for the Artosphaera maxima in dry and frozen biomass. Meanwhile, our objectives are first, to count the cell number to get the growth curve. Second, to analyze the protein content. Third, to measure the pigment content. And fourth, to observe the antioxidant activity. Here we use Artosphaera maxima. The Artosphaera maxima stock of each media was transferred into new media as pre-culture. After 7 days cultivation, 200 milliliter was transferred into new media to get 1000 milliliter culture. After 7 days of cultivation, 1000 milliliter culture was transferred into new media and total 5000 milliliter culture was cultivated. During cultivation, we observed the growth by counting the cell number. And after 14 days, the biomass was harvested and collected for further treatment. The collected biomass was processed. First, the biomass was dried in the oven at 50 degrees Celsius for 24 hours. After we got dried biomass, the biomass was pulverized into fine biomass and kept in dry. Second, the biomass was kept in freezer. Then, the dried and frozen biomass was diluted into double distilled water and extracted by freeze-thaw extraction. The biomass was frozen for 2 hours at minus 80 degrees Celsius and thawed at around 30 degrees Celsius for 24 hours. After centrifugation, the supernatant was collected as cell-free extract and had been analyzed for its protein content, pigment content, and antioxidant activity. Here are the results. For the growth curve, we measure the cell number by using hemocytometer. With magnification 100 times, we count the cell number. As you can see at the figure, freshwater xerox medium produces 550,000 cells per milliliter with 1.1 gram dry biomass. Meanwhile, seawater xerox medium only produces 460,000 cells per milliliter with 0.9 gram per liter media. This result suggests that the biomass production in freshwater xerox medium was slightly higher compared to the seawater xerox medium. For protein analysis, 60 well plate was used and cell-free extract was added into well plate and reacted with Bradford reagent. The absorbance was observed using virus scan at 595 nanometer and bovine serum albumin was used as control. Protein concentration in the cell-free extract of dry and wet seawater biomass were 0.18 and 0.19 milligram per milliliter. Meanwhile, dry and wet for freshwater biomass were 0.28 and 0.37 milligram per milliliter. Here, we also showed the protein content in each biomass. The protein content in the biomass was smaller than theoretically. It might be because the freeze-thaw process in this study was not efficient in extracting the whole protein content. This result showed that the wet biomass of freshwater xerox medium had the highest protein content in its cell-free extract. For pigment analysis, the cell-free extract from each sample was analyzed for its absorbance at 560, 620, and 652 nanometer. Using this equation, the result showed that wet biomass of freshwater xerox medium cell-free extract had the highest total pigment content, about 189 microgram per milliliter. If we observe more, the wet biomass cell-free extract had a higher pigment content compared to dry biomass in each media source. And previous results showed that the dry biomass from freshwater xerox medium had higher protein content than wet biomass of seawater xerox medium, but had a lower pigment content. This result suggested that the drying process and its storage could decrease the pigment content, or it might be because the frozen storage could prevent the deterioration of the content. For antioxidant activity, the cell-free extract was analyzed in 3 different concentration values by diluting the cell-free extract solution with double distilled water at 1 time, 2 times, and 4 times, and reacted with ABTS reagent. The sample solution absorbance was analyzed by virus scan at 734 nanometer. Vitamin C was used for antioxidant standard. The results showed that the most potent sample for antioxidant activity was dry biomass from seawater Zarup medium with the lowest IC50 at 3.8 milligram per milliliter. Meanwhile, IC50 value of vitamin C was 27.7 microgram per milliliter. Compared with the standard, the antioxidant activity of the cell-free extracts was still low. However, the dry cell-free extract had higher antioxidant activity than wet biomass. This result suggests that the antioxidant properties other than phycocyanin were more concentrated in dry biomass through the drying process and storing process, making it more soluble during extraction. Here we showed you some documentation during our study. As summaries, the biomass production in freshwater Zarup medium was slightly higher compared to seawater Zarup medium. Second, in this study, the wet biomass of freshwater Zarup medium had the higher mass protein content in its cell-free extract, which is 0.37 milligram per liter. Third, dry biomass from freshwater Zarup medium had higher protein content than wet biomass of seawater Zarup medium, but had a lower pigment content. The drying process and its storage could decrease the pigment content, or it might be because the wet storage could prevent the deterioration of the content. Fourth, the dry cell-free extract had higher antioxidant activity than the wet biomass. This result suggests that the antioxidant properties other than phycocyanin were more concentrated in dry biomass through the drying and storing process, making it more soluble during extraction. The last but not least, we would like to thank to BRIN for the funding. Thank you very much for your kind attention.
Thank you. For the next speaker is Mr. Rian Haryo Setiawan, will present entitled The Hidden Superfood, Microalgae from Indonesia and its Potency as Functional Food Ingredients. Please play the video.
Hi everyone, my name is Ryan and today I'm going to present my presentation titled The Microalgae from Indonesia and its Potency as Functional Food Ingredient. And here is the outline of my presentation. So first I'm going to talk about the biological aspect of microalgae and how to isolate, cultivate, and produce it. And after that, I'm going to explore the nutritional and functional property of microalgae. And in the end, I'm going to give some instance of application in the Indonesian functional food product. So Indonesia have been suffering for double burden malnutrition. It is a condition where undernutrition and overnutrition coexist at the same time. And this condition will affect the Indonesian human resources quality because it's lowering its productivity and its health development. And in the end, it will affect the national economy because of that. A lot of factors influencing this double burden malnutrition, and one of the most major is poor diet choice. Therefore, the pursuit of novel and highly nutritious and healthy source of food become one of the most favored solution. Indonesia, with its huge aquatic territory and huge exposure to sunlight throughout the year, has created a great microalgae biodiversity. And this microalgae can be bioutilized into a lot of prospects from energy, agriculture, pharmacy, and functional food. And Therefore, the aim of this paper is to review the potency of microalgae that is isolated from Indonesia or deposited in culture collection in Indonesia as a functional food ingredient. So, microalgae has a microscopic
Excuse me, Mbak Selin, the video is stopped.
vision based
Oh, yeah,
on
okay.
its taxonomic classification. And to grow, microalgae need the supply of carbon dioxide and lightning as their source of energy. And because they produce oxygen as their metabolite, it has to be removed. And temperature and mineral affecting their growth as well. And to isolate microalgae, we can use the conventional methods like culturing on agar like any other microorganism. But the more advanced technique has been developed by using the flow cytometry approach. But of course, it needs an advanced instrumentation and also highly cost. And to cultivate, they can be grown in a multi-stage of cultivation with gradually increasing its volume. And sometimes in each stage of its cultivation require a specific condition for specific microalgae. Meanwhile, for the large-scale production, there are actually two schools of thought of system of large-scale production: the open and the closed system. Well, for functional food, the closed system is preferred because it can control well, and it can avoid to get a contamination from a foreign material. While for nutritional properties, a lot of microalgae from Indonesia has been explored for its nutritional properties. And one of the most interesting part is that different kind of microalgae have a different content as well. For example, in the protein content, a different kind of microalgae have a very different protein content. And not only its nutritional content, they're affected by their type, it's also affected by where they are isolated. For example, in the mineral content of same type, Spirulina platensis, they have different mineral content because they are isolated from different saltwater and freshwater. And for functional properties, one of the most explored its functional properties is antioxidant. So a lot of microalgae have been extracted using different kind of solvent to explore its antioxidant activity, and also it has been explored to see its antimicrobes against a lot of pathogen. And these two characteristic of microalgae can not only it's beneficial for the health of human, but also if it's applied in food, they can prolong its shelf-life because oxidation and microbes is two major cause of food spoilage. And also some microalgae have been explored for its anti-cancer activity and its pigment. Well, microalgae consists of two groups of pigment: the carotene that consists of beta-carotene, chlorophyll, and also phycobilin protein that consists of phycocyanin, phycoerythrin, and etc. And this pigment is not only beneficial for the health of the body, but also it can increase the quality of the food because it can become a food colorant. And some microalgae have been also been explored for its anti-hyperglycemic activity by looking at its activity in inhibit the alpha amino acid or the enzyme that digesting carbohydrate in a human body. And its application in various functional food can be found in various functional food. For example, in savory food, some microalgae have been fortified into a fish sausage, and it's not only it's found that by fortification of the microalgae is increasing its texture. and giving a better sensory acceptance. And they can also be applied in bakery product. And it is found that microalgae can increasing its nutritional properties, its health effect, and prolong the bakery product shelf life due to its antioxidant and antimicrobe activity. And in ice cream and beverage, well, microalgae can be also applied in this type of food, but too much microalgae can give off flavor because they have an off odor. Therefore, they need some extra treatment to mitigate this problem. And also, they can also microalgae can be used for the health supplement. They can be used for supplement for pregnant lady because of its antioxidant and essential amino acid and omega-3 content. They can also be used as church and lozenges. So in conclusion, microalgae has a potency as a novel food ingredient to mitigate double burden malnutrition in Indonesia. And the diversity of type, isolation site, and growth condition give distinctive properties of each microalgae. Therefore, they need the deep characterization and screening. And culturing optimization and large-scale production is needed to make sure the accessible of microalgae to the population. Thank you.
Thank you very much. The last speaker is Miss Marlene Megalestin Raunsay. Will present entitled Bioprospection of Endophytic Fungi associated with plants from Central Halmahera, North Maluku as antibacterial and antioxidant agents. Please play the video.
Thank you, moderator, for the time. Good afternoon, ladies and gentlemen. I am Marlene Megalestin Raunsay, will present our study entitled Bioprospection of Endophytic Fungi associated with plants from Central Halmahera, North Maluku as antibacterial and antioxidant agents. I'm Marlene and my team, Mrs. Kartika, Mrs. Praptiwi, Mr. Muhammad Ilyas, and Mr. Andre Agusta. We are from National Research and Innovation Agency. Endophytic fungi live in the host plant's tissue and spread throughout the host organs, including leaf, stem, root, fruit, flower, and seed. Endophytic fungi hold essential roles for their host plant, for instance, through promoting plant growth by producing phytohormones and creating competition with the pathogen. The ability of endophytic fungi to produce bioactive compounds is still beneficial for their host plant and also advantageous for drug discovery. Endophytic fungi also have been reported to mimic the host plant metabolites. For example, the ability of endophytic fungi associated with taxol-producing plants to produce taxol. Until now, the exploration of endophytic fungi as potential sources of active compounds is still limited, especially endophytic fungi associated with Indonesian plants. In this study, we screened the antibacterial and antioxidant potential of 24 isolates of endophytic fungi associated with plants collected from Central Halmahera, North Maluku, Indonesia. This table shows that there are 24 endophytic fungi isolated from eggplant species from Halmahera Island, and endophytic fungi were mostly isolated from leaves and some from rhizomes. And these are the methods used in this study. There are 8 steps. First, an isolation of endophytic fungi. For example, Polycias coolsey and Morinda umbellata. The healthy plant parts were washed thoroughly in running water, and the surface sterilized using 70% ethanol for 1 minute, 5% sodium hypochlorite for 3 minutes, and 70% ethanol for 30 seconds, and rinse using sterile distilled water. And dried sterilized samples were cut into fragments, placed on top of the CMMA supplemented with chloramphenicol 0.05 gram per liter, and incubated at room temperature for a week. And the colonies were isolated, purified, and maintained on PDA. And for identification of endophytic fungi, based on the morphology approach, from macroscopic and microscopic characterization using these parameters. And cultivation and extraction of secondary metabolites. The culture were incubated at room temperature under dark and still condition, and after 3 weeks the biomass and the media were extracted using ethyl acetate for 3 times, and will be extracted use liquid extraction, and the extract were concentrated using a rotary evaporator. And for chemical compounds analysis, used thin layer chromatography method, and the TLC plate will be visualized under UV light, and followed by spraying with vanillin sulfuric acid and cerium sulfate. And for antibacterial test using TLC direct biochography method, and then the TLC plate will be dipped in the bacterial suspension of S. aureus and E. coli, and after incubation will be sprayed with INT, and the white zones indicates the presence of antibacterial activity. And then followed by determination of MIC, used microdilution method, that the microwell plate will be incubated. The sample in the microwell plate will be incubated, and then we can know the MIC values. And for the antioxidant test, we'll use the TLC direct biochographic method. The TLC plate will be sprayed with DPPH, and the yellow color font indicates the presence of antioxidant activity. And for quantitative determination of antioxidant activity, the microwell plate will be incubated in the dark place and for 90 minutes. And after that, will be measured absorbance at the wavelength of 517 nanometers, and would be calculated use this formula. And the result and discussion. This is the figure of the microscopic view of representative fungal endophytes. The fungal strains and identification through morphological observation showed that selected fungal endophytes belonging to the taxa Aspergillus, Coletotrichum, Lasiodiplodia, Neocosmospora, Pomopsis, Trichoderma, and Silaria. In this study, endophytic Coletotrichum dominated the obtained strains, where 29% strains of them were isolated from 2 species of medicinal host plants. And this is the chemical profile analysis. Figure 2 shows the similarity of chemical profiles in several extracts. For example, for N5, 10 and 11, and 9 to 23 belonging to the genus Coletotrichum. And for antibacterial biochography, the dot plot biotogram against S. aureus showed antibacterial activity at sample numbers 1 to 5, 10 to 17, and 22 to 24. The extracts that were active as antibacterial against E. coli were 1, 2, 4, 5, 8 to 17, 21, and 24. The selected extract were then eluted and tested against S. aureus and E. coli. And for antioxidant TLC biochography, figure 5a shows that all extracts had qualitative antioxidant activity, And the distribution of antioxidant active spots with various retention factors can be seen in figure 5b. Based on figures 5, the determination of the IC50 values was focused on 9 extracts. And this is the complete table of bioactivities of endophytic fungi. Several extracts show higher MIC values. For example, MIC value of MUHD1 is 128 microgram per milliliter, ADHD1 and PSHD4 are 512 microgram per milliliters, again in some range. And GLHD3 has an MIC value of 512 against E. coli. And for antioxidant activity, moderate antioxidant activity was indicated by extract codes DMHD2 and MUHD1 with IC50 values of 85.43 ppm and 64.8 ppm respectively. And the conclusion, the antioxidant evaluation in this study displayed that 2 endophytic fungi extracts showed moderate antioxidant activities, and in addition, 3 extracts of endophytic fungi had moderate antibacterial activities against S. aureus and 1 extract against E. coli. This finding indicates that endophytic fungi is isolated from plants collected from Central Halmahera, North Maluku, are potential as antioxidant and antibacterial agents. Further studies need to be carried out to isolate the bioactive molecules responsible for these activities. And this is the acknowledgements and the reference. Thank you.
Okay, thank you for the videos. And now it's time to discussion session. Please, the participants who want to give some question, you can type on chat or you can raise hand. Okay, Mbak Sanli, you raise hand.
Yeah.
Yeah. Okay.
I have a question to Ms. Marlene Ramsay regarding the local plant Polycia and Morina. That what what were the local people use for the local plant in Halmahera? I mean, you mentioned that this plant for medicine, right? I was wondering that this plant for what?
Okay, thank you, Miss Mbak Sanli. Miss Marlene, okay, you can respond.
Okay, thank you, Kak Senli, for the question. I didn't mention the function in the articles, but for I know that the medicinal plants are used for like malaria, and then some plants for diabetes, and then some extract for some plants also from for like for the mom. So meriang. Meriang. So but I didn't put in my articles. I just focus to screen. We just focus to screen the extracts. Thank you.
Okay, thank you, Miss Marlene. For the next, okay, Mr. Yan, you can say your question, please.
Yeah. Hello. I want to ask to Mr. Baso. I want to ask about, in your opinion, what's the reason why different stage of microalgae have a different pigment and antioxidant activity? I mean, why? Thank you.
Okay, thank you. Mr. Baso, you can respond it?
Yes.
Okay, please.
Can you hear my voice?
Yes.
Yeah. Okay, in my opinion, actually, the pigment concentrations in microalgae, like for instance SP, is affected by many factors, but in the relations with with its what is it growth phase is because the biosynthesis of the secondary metabolites in this organism actually usually produced when they enter the is it stationary phase, because or maybe the end of the logarithmic phase, because most of organism actually use the secondary metabolites to defend their self, or maybe it's not essential for their growth actually. So I think the more maybe it's one of factors that that affect concentration difference in every phase, growth phase. I mean.
Okay. Mr. Baso, so if we want to harvest chlorophyll and also carotenoid, which time you will suggest?
Yeah, it's actually different. If you want to harvest the carotenoid, then it should be harvested in stationary phase. But
in stationary
if you
phase,
want to, yes, in stationary phase. But if you want to, what is it? Harvest the chlorophyll, the highest chlorophyll content, you should harvest it in. You can in log phase or in stationary phase, because based on our experimental result, we found that log phase and stationary phase didn't the content of chlorophyll didn't different statistically.
Okay, thank you, Mr. Baso. Okay, next question. I will read from chat box from Mr. Hairul Anam to Mr. Haryo Bimo. Thank you for your nice presentation and information about Champollion. Could you please kindly explain the tool that you use for organoleptic and sensory tests?
Okay.
Please. Yeah, okay.
Thank
Please,
you
Mr.
very
Haryo.
much, Pak Anam. I will explain about the methods to organoleptic test and hedonic test for Velva Champollion products. So the organoleptic test and sensoric test, you know that sensory testing is a way of testing using the sense of humans as the main tools to see the quality of food products that have undergone processing. The hedonic test is a method used to measure the level of preference for the product using an assessment sheet or questionnaire in determining the quality level based on a scale of 1 as the lowest and 9 as the highest response for color, aroma, texture, taste, and overall. The scale used in 9 is very much like, 8 very much like, 7 like, 6 somewhat like, 5 neutral, 4 slightly dislike, 3 is dislike very much, 2 very much dislike, 1 is very much not like. Then the hedonic test was carried out for 30 semi-trained panelists. If you use the trained panelists, it's just for 10 panelists. If the trained panelist is just 10, but for semi-trained panelists is needs sample for 30 questionnaire. Thank you very much.
Okay, thank you. Thank you, Mr. Haryo. Mr. Hairul, okay for the answer?
Okay, thank you for your explanation.
Okay. For the next question. Okay, Miss Marlin, please.
Okay, thank you, Mrs. Wenning. I want to ask Rian. There is a interesting presentation. I want to ask you, based on your reviews, what parameters most affect the nutritional content of microalgae and why? Okay.
Okay, Mr. Yan, you can respond.
Yeah, thank you for your question. So for our review that I have collected from various literature, that it is found that different type can give distinctive nutritional properties. I believe that different type or different species have a different DNA, and it will affecting how the microalgae living, how their physiology, and then that's one factor that affecting their nutritional properties. Another factor that affecting is their external factor, the presence of the salinity, such as the salinity that also have been proven in the Mr. Khairul Anam's paper, that it affect the nutritional quality, nutritional property of the microalgae. So that's my answer: the internal factor, the DNA, and the external factor, the way where they are lived. Thank you.
Okay, Mr. Yan. One question from me. It's good to see that the superfood, you say superfood microalgae already used and commercialized in Indonesia. Maybe you have experience. Maybe you can suggest one or two products, maybe from microalgae that you already know, good for healthy or maybe for supplement.
Yeah, actually, you can find a lot of various product of that using microalgae in the market. You can find in your Tokopedia or your online e-commerce. You can find that, and but the one that I put it in this review is the only that food application that using research. For example, like we want to see the effect of the macroalgae, but usually in the market, I found that a lot of food product using the only one species, the spirulina or
spirulina
chlorella. Spirulina or chlorella.
For supplement.
Yeah, exactly. And also there's also potency to be used as a supplement for protein, because of their protein content. They can be used for the source of protein.
Oh. Yeah. Okay, thank you for the information, Mr. Yan. For the next question, we still have time. Okay, maybe I will ask to who is Maxima, to Mr. Khairul Anam. Okay, you say about spirulina maxima, right? But I wondering why you interesting to spirulina maxima beside the information still limited. You just say the information still limited, but maybe you have another reason for that. And second, in summary, you mentioned about the dry biomass and wet biomass. From your research, if it goes to a larger production, what do you suggest to use, dry biomass or wet biomass? Okay, please, Mr. Khairul Anam.
Okay, thank you very much for your kind question. I will try to answer the first question, why I use spirulina maxima. Here I use spirulina maxima because we are obtaining by ourselves in our lab from Semarang, from the sea of the northern part of Java. So
it's local from
local
Indonesia.
Local strain of Indonesia. So if we want to develop in collaboration with the industry or else, we can freely use this strain as commercial product. And then for the second question, dry biomass or wet biomass, if we use dry biomass, we using the oven, and for the wet biomass, we are using the freezer. Actually, from my research, that the dry biomass contain the antioxidant activity from the dry biomass is higher compared to the wet biomass.
Yes.
The difference with the wet biomass is higher in the pigment content. So if we use the Spirulina maxima for the antioxidant activities, for its antioxidant activity, maybe the dry biomass still okay, and maybe more cheaper compared to the wet biomass, because we have to keep in the freezer. Maybe that's my answers.
Okay, thank you, Mr. Hairul Anam. Okay, other participant will give some question for the speakers. Okay, I think maybe I think the time is already over. Thank you for all the speakers and also the participants. I think this session is good because the question is a lot. Yes. Okay. So it is the end of this session. Once again, thank you. Keep spirit, stay healthy, and good luck for your research and also your project. And see you in the next event. Wassalamualaikum warahmatullahi wabarakatuh. Please, yes, please note from Mbak Sally, all participants should back to main room. So because there are several interesting presentation, so you can press the leave room to back to the main room. Okay, thank you for everyone.
Thank you very
Bye.
much.
Thank you.
Thank you. Bye.
Bye.
Recording in progress.
Welcome back to the main room, ladies and gentlemen. Now, we would like to invite Ms. Novio Ayusril, Product Specialist, Protemix Nutrilab, to share the presentations. Ibu Novia, good afternoon. Thank you for joining with us. Can you hear my voice?
Hello.
Yes, time is yours.
Thank you, everyone. Let me give me one moment, please. Yes. Do I need to present in English or should I just in Bahasa?
It will be better in English, Ibu, because the participants not only coming from Indonesia. Thank you.
Okay. Thank you. Yeah. Good afternoon, everyone. My name is Novio. I'm a product. Nutraceuticals is one of distributors bio life science products in Indonesia. So our office is in Kebayoran Lama, South Jakarta, and here you can see you not only can find us in Jakarta, but we also have office like branch office in Bandung, Semarang, Yogyakarta, Surabaya, and Bogor. So for BRIN especially, we have ourselves here, which will help you guys whatever you need from us. So you can see in the right side that we can support you guys with these products, but mainly for Nutrilab, you can reach to us for Gitco product, Invitrogen, and Thermofisher scientific products. This one especially for you guys who are doing biology or biomolecular research and also cell culture research. And also, not only that, we have several products which support your like general instruments such as centrifuge, cortex, etc. So we have also bio-based and also Hanil from Korea and Clever Scientific from UK. For the plastic consumables, we have products such as CPP, Tarson, Nest, etc. So this is the short introduction from me for our company, and maybe I will move to the next slide about what is the product or what can we do or what can we help you with your research here. So we have several divisions in our company, mainly supporting this group of research. First one is molecular biology. You guys who use PCR reagent or like real-time PCR, and also our best seller product is qubit, come from Invitrogen product. And also for protein biology, such as antibody, ELISA, Western blot product, especially for this product line, you can reach out to me if you want to have more information about protein biology. And then we also support you guys with synthetic biology from Thermofisher. If you're doing expression protein and also cloning, you guys can reach out to us how to order, or you want to try like positive control for your real-time PCR, you can also reach out to us. And then cell biology, our main product, which is from Gitco. So we are the only one, like the only authorized distributor for Gitco product line in Indonesia. So if you are doing cell culture, you can also reach out to us. And then the last one is cell analysis product, which is microscope, fluorescent microscope, flow cytometry, and also high content screening. This one is the for protein analysis flow. As I mentioned before, that anyone who use protein like to do such analysis like Western blot, mass spectro, we have these many products that can support you guys. So from the extraction of the sample to the protein cleanup like dialysis, desalting, concentration, and also the downstream applications such as I mentioned is the stage Western blot workflow. We have here like Western workflow that maybe you guys also have heard before, that usually you takes 2 days to do Western blot, right? We have product line that can use that you guys can use only in 4 hours. You can get the result. So if you guys are interested, you can reach out to us for this application. And the last time is the analysis. Next is PCR workflow. As you can see here, we have we can support you the whole workflow from the extraction of DNA or RNA and then the quantitation. So here you guys may be familiar with nanodrop or spectro. So here we have a qubit fluorometer which use fluorometer detection, and also we have the master mix for both conventional and real-time PCR, and then of course the PCR instruments, and also the electrophoresis system here which use power snap. Next, as I mentioned, that we support you guys with the GIPCO cell culture media. Maybe you are familiar with our GIPCO product already. So you can see that from the isolation and then to the storage media, you can see here that we can support you guys with this. This one is our like high-end product, which is high content screening. Maybe you are familiar with this this term, right? That you can use this microscope, fluorescent microscope light plus microplate reader, and also that analysis data for your drug discovery. So you can see that this, for example, you have like a chemical compound that targeted RNA or other target in your cells, and then you want to try to check whether this drug is actually works for your cancer or for other diseases. So you can try with our high content screening cell infected CX7 LBL Pro. And then maybe for the next level, you can also image the this drug with bioluminescence. This is, I think, my last two slides for our general equipment, such as I mentioned before, right? The freezer, centrifuge, like UV cabinets, and also all the stuff that you are familiar with in your lab. You can also, if you want to purchase your unit, you can reach out to us, or maybe some consumable or like accessories that you need, you also can reach out to us. I think this is my last presentation. So this one not only for bioscience products, we in Nutralab also support you guys with bioproduction products. Maybe next time, now you are doing like the R&D step, right? And maybe in the future you want to produce your own like vaccine or your own drugs or protein recombinant. We can support you guys with these product lines from research development, bioreactor or fermenter. Also, we have this for biofactoring. Maybe you guys doing cell culture, which like adherent cells. You can also use our biofactory from next. And the last time is for purification of your protein in large scale. Usually you just only use like thin column or ASTA, right? You're familiar with that, those stuff. Here we have the resin, which can help you guys to use it in your purification system. Maybe that's all from me. This is I'm very grateful for this opportunity to present to you guys all our products in Nutralab Pratama. So thank you so much. Yeah, see you guys.
Thank you very much, Ms. Novio, for comprehensive explanations. As what mentions, ladies and gentlemen, you can contact Nutrilab directly for further informations of their services and also their products. So, distinguished guests, ladies and gentlemen, as the time is now 12:22, we will have a break for approximately 1 hour and 8 minutes, I guess. And please kindly back to the Zoom meeting at 1:30 p.m. Jakarta time. Once again, we will have a break for approximately 1 hour and 7 minutes from now on. So please kindly back to the Zoom meeting at 1:30 p.m. Jakarta time. Thank you and have a nice lunch.
Hello, Adel.
Hello, Uni.
How are you?
Hey, fine, fine. How are you?
Very
Good to
good.
see you.
Yeah.
Great, great.
Yeah. So what's your? Are you already okay now?
Yes, I'm fine. Yes.
Okay. Okay. So how long?
Shall I just
Yeah. Start your
share my screen?
Yes. Yes, please.
Because I'm trying to do that with two computers, so I have to see how this is working. Just a second.
Yeah.
Stop sharing, because
Enggak, share screen. Are you familiar with Zoom, Arnold?
I'm sorry?
Are you familiar with Zoom?
No, we are normally using mostly Teams.
Teams. Oh, okay.
But I try to share my screen.
Yeah.
But I cannot. But I get the message. You cannot start screen sharing while the other participant is sharing.
Okay. So the organizer will stop this. Okay. Now it's already okay. You can
now it
try.
should be possible.
Yeah.
Okay. Let me just try. Yes.
Yeah, I already see your presentation.
Yes, but there's still some annoying thing in here.
No, it's
Let me check. There it is.
Yes.
Yeah, it's working. Perfect.
Okay.
Good. Now, I'm glad it's working, Uni. So this is the way we will do it.
Yeah, Arnold. So after the break, around 6 minutes before your talk, there will be a parallel session, and you can choose which room do you want to.
Yeah, I will check.
Yeah.
I want to go.
Yeah.
But just a second, I'm trying to get out of the screen sharing now.
Oh, gimana caranya kamu?
Because that's not working yet.
Ya udah. Gimana caranya kalau mau keluar dari share screen?
Here.
Yeah. Share. So in this
I'm still sharing, eh? Or not?
No. You already stopped sharing your screen.
Okay, then it's fine.
Yeah.
All right. I guess it will work.
Okay, thank you very much, Arnold, for your participation.
No, I'm very happy to do that. Thank you for the invitation. It's nice.
Yeah. In this afternoon, there will be only you presenting in the plenary. Yeah, plenary speakers.
Yeah.
So you will have a lot of time for presentation and the discussion. Yeah.
Okay, fine. Yes. Great.
Okay, I'll not see you later.
See you later then. Okay.
Yeah.
Bye.
Good afternoon, ladies and gentlemen. Welcome back to the NINF of ISIBio. The next agenda is continue the parallel sessions. Also, please kindly back again to the room, and you can choose the room based on your interests. Allow me to read the 5 rooms today. Room 1 with the theme of bioresources and biodiversity. Room 2, Nutraceuticals and natural products. Room 3, biotechnology and bioinformatics. Room 4, environmental sustainability. And room 5, biorefineries and circular economy. Each room will be led by moderator, so please kindly click the feature of join breakout room. You can find out on the left corner of your Zoom, there will be a more and then join breakout room. 3 dots more and then join breakout room. Once again, the breakout room will be divided into 5 with the different speakers, also with different themes. Room 1 will be bioresources and biodiversity. Room 2 will be nutraceuticals and natural products. Room 3, biotechnology and bioinformatics. Room 4, environmental sustainability. And the last room, biorefineries and circular economy. We hope you enjoy the sessions. Thank you.
Wabarakatuh. Good afternoon, everyone. I would like to thank you to the Committee of ISIBio 2022. Here I will present our research with the title Evaluation of Antibacterial and Antioxidant Potential of Ficus spp extract from Simelu Island, Aceh, Indonesia. I'm Evana and this is my team, Dewi Wulansari, Ahmad Fatoni, Praptiwi, Marlin Megalestin Raunsai, and Andrea Agusta. We are from the National Research and Innovation Agency. As we know, a global health issue related to antibiotic-resistant bacteria continues to increase every year and has an effect on the economy because of high medical costs. Inappropriate use of antibiotic is one of the causes of antibiotic resistance. On the other hand, the problem of free radicals is also a major issue related to the emergence of various degenerative diseases by attacking human biological molecules. The discovery and development of antibacterial and antioxidant agents from nature have a high potential in terms of Indonesian natural wealth and the side effects of their use. Higher plants have long been used as traditional medicines to treat human diseases. Approximately 80% of people worldwide use plants as safe source of medication to cure human diseases. Ficus is one of the available indigenous medicinal plants in Indonesia. Is a member of the Moraceae family. This genus is known to have efficacy as traditional medicine in several regions in Indonesia. Some of the Ficus species have biological activity such as antioxidant from Ficus deltoidea, Ficus auriculata, and also Ficus racemosa, as well as antimicrobial activity from Ficus cordata and Ficus benghalensis. Biological activity assays of other ficus species need to be done because of their great potency for human health. Exploration of several species of ficus plants was carried out in Simelu Island, Aceh, Indonesia. The focus of this study is to evaluate the antibacterial and antioxidant activities of 4 plant species of ficus such as Ficus fulva, Ficus rips, Ficus septica, and Ficus sp. In our study, 4 plant species of ficus we collected from Simalu Island in 2017. These plants were extracted by graded maturation method using flow solvent based on increasing polarity solvent from non-polar solvent to polar solvent. A total of 32 extracts we analyzed for antibacterial activity against Staphylococcus aureus and E. coli, as well as antioxidant activity against DPPH free radical. The antibacterial and antioxidant assay we carried out qualitatively using the TLC bioautography method, and the active extract were then analyzed by quantitatively to evaluate the minimum inhibitory concentration and IC50 value in 96 microwell plates using the microdilution method. And this is our result. Based on the result, the percentage of yield extract varied from 0.167 until 4.062%. It showed that the chloromethan extract of ficus ribs fruit showed the highest extraction yield, while the lowest yield was obtained by ethyl acetate extract of Ficus septica stem. The variation in percentage yield of extract can be attributed to the extraction solvent, the composition of the extract, the plant parts, the maturity of a plant part, season, and agroclimatic condition. The TLC chromatogram profile of extract we detected under 254 and 366 nanometer UV light, followed by vanillin and cerium reagent spraying to evaluate the chemical compounds of the extract. The varying retention factor of each spot produced in TLC chromatogram profile of the Ficus SPP extract with different solvent demonstrate that this extract contain distinct chemical compounds. The compounds that have the same physical properties as the stationary phase, we use silica as the stationary phase, will be retained longer in the stationary phase because of the high polarity of the silica compounds. The more polar compounds interact stronger with silica and eluted later, resulting in a lower retention factor value, while the less polar compounds will be eluted first. And this is the result of antibacterial activity. A TLC bioautography method was used to perform antibacterial activity screening against S. aureus and E. coli. We used chloramphenicol as a positive control. Inhibition of bacterial growth is indicated by the appearance of a white zone surrounding the extract on a purple background. The purple background on the TLC plate was caused by the reaction of dehydrogenase enzymes in living bacteria converting INT to the purple formazan. Based on the result, it showed that 26 extract inhibit the growth of S. aureus and 29 extract inhibit the growth of E. coli. And then this active extract were then analyzed by quantitatively to evaluate the minimum inhibitory concentration value. Based on the MIC value of the extract, it showed that 6 extracts had strong antibacterial activity with the MIC value of 64 microgram per milliliter. 1 extract from ethyl acetate extract of Ficus fulvalis has strong antibacterial activity against S. aureus, and 5 extract from ethyl acetate of Ficus septica leaves, methanol extract of Ficus septica leaves and stem, ethyl acetate and methanol extract of Ficus sp leaves. They have strong antibacterial activity against E. coli. The difference in antibacterial activity of the extract is influenced by parts of the plant and solvent's capability to extract antibacterial compounds. Chemical compounds with antibacterial activity may be attracted to more polar solvents such as ethyl acetate and methanol rather than less polar solvents such as N-hexane and dichloromethane. In ethyl acetate and methanol extract, leaves extract is highly effective than the others. The antibacterial activity of methanol extract of leaves of Ficus species because of the presence of biological active compounds such as tannin, terpenoid, alkaloid, saponins, flavonoids, essential oil, and phenolic compounds which are present in significant amounts. And this is the result of antioxidant activity. Based on the result, it showed that 14 extracts have antioxidant activity. We use catechin as a positive control. The yellow spot against the purple background after the PPH reagent spraying reveal antioxidant activity. The intensity of the color indicate the antioxidant capacity of the extract. And then these active extracts were then analyzed by quantitatively to evaluate the IC50 and the antioxidant activity index values. Based on the classification of antioxidant activity index, methanol extract of stem of ficus ribs saw the highest antioxidant activity. The stem of ficus SPP extract exhibit higher antioxidant activity in comparison to other parts such as fruits and leaves. The polarity of the solvent affects the antioxidant activity. It showed that methanol extract produce higher antioxidant activity. Ficus species have been identified to be a rich source of polyphenolic compounds such as gallic acid, caffeic acid, and chlorogenic acid, and flavonoids such as quercetin, catechin, and chimeferol, which contribute to the high antioxidant activity. And this is our conclusion. The antibacterial and the antioxidant activities of ficus SPP extract were affected by plant parts and solvent for extraction. More polar solvent attract more chemical compounds with antibacterial and antioxidant properties. Ficus SPP extracts in ethyl acetate and methanol extract inhibit the bacterial growth more effectively than other solvents. The highest antioxidant activity was also shown by the methanol extract. Leaves extract to be more active than other parts in inhibiting the growth of gram-positive bacteria, S. aureus, and negative bacteria, E. coli, while the stem extract are more effective than other plant parts in scavenging the PPH free radical. Ficus SPP extract are promising natural sources of antibacterial and antioxidant agents. The bioactive compounds of ficus SPP extract should be isolated and purified for further study. Okay, I think that's all of my presentation today. Thank you so much for your attention.
Okay, thank you very much, Ms. Ivana, for your presentation. And then the next, we will continue with the second speaker, Ms. Amalia Nur Fauziah. Please, Ms. Amalia.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. My name is Amalia Nur Fauziah. I'm the presentation with the title of paper, Effect of Preparation Method on Moringa leaf extract, Nano-emulsion and Focal. Moringa leaf salam, or moringa plant are also referred to as the miracle tree because all parts of the moringa plant contain nutrients that can be utilized. The leaf contains flavonoid, polyphenol, lycopene, and beta-carotene. The main flavonoid in Moringa oleifera is quercetin. Propolis is resinous texture that honeybees from Trigona sp. From the bark of the trees, they consume and in use to protect the hair from fungi and bacteria. And for nano-emulsion are referred to as mini nano-emulsion. A nano-emulsion is finely dispersed oil and water emulsion system that has the precise range 15 until 100 nanometers. Unlike microemulsion, nano-emulsion are kinetically stable. Nano-emulsion can also be formed in various formulations such as foam, cream, liquid, and spray. Nano-emulsion are used for drug delivery system and cosmetics. First, methodology. Water phase and oil phase. Before homogenize using ultrasonic, water phase sonication for 1 minute. Water phase and oil phase homogenizer with ultrasonic. Pasteurizing homogenizer with variation in spring, speed of 300 rpm, 600 rpm, and 1200 rpm with time 5 minutes. After a thread using ultra the all motion were followed by a thread using ultrasonication for 50 minutes. The non-emission was found and can be analyzed with some analysis such as particle size, stability, and Microsoft Digital. Result and discussion. On this page, we can see that the antioxidant activities from morning leaf extract and propolis. In the table, antioxidant activity of morning leaf extract and propolis, that the acidity of morning leaf extract and propolis has less than 100 ppm, that can be categorized as a strong antioxidant. Use the result of LCMS from morning leaf extract. The mass spectrum captured at a specific retention period, that the camphorol compound was visible at molecular weight of 595 and 449 with retention time of 2.78 and 3.6, while compound contained quercetin were visible at molecular weight of 465 with retention period with 3.29. This is the result of particle size analysis. From the table, we can conclude that the sample care in table 2 with the smallest particle size, which is HLB or hydrophilic, lipophilic 9 with 97.5% water-based with an average particle size of 96.03 nanometer and a polydispersity index value of 0.36. The standard deviation between the sample size and average graphite size, (OT), demonstrates the uniformity of the graphite size in some. A more homogeneous particle size distribution will be produced with a lower peak value. Addition P is one that is less than 0.5. The value indicates that the goal distribution is not uniform if it's more than 0.5. Since the sample can meet the standard, it can be claimed that the sample can belong to the category of optimal non-emission. This is the result of stability of the 9 sample non-emission that with observation after a 6 weeks, which reveal that they don't exhibit any noticeable change and generally tend to be stable. This demonstrates that the components of the produced nano-emulsion are the proper concentration to prevent precipitation, creaming, and separation. If the nano-emulsion effective, if it doesn't form layers and doesn't change in color or consistency, it is said to be stable. This is the result of Microsoft Digital. The figure demonstrates that the particle size of the 97.5% water basis with a homogenization speed of 600 rpm is smaller than the other sample of nano-emulsion. This support can be claimed from the particle size analysis, which show a value of 196.03 nanometer and a P of 0.361. Conclusion: based on this research, we can conclude that the formulation show the optimum nano-emulsion were sample K. It consists of 97.5% water phase, consists of marine olive extract, propolis, DN80, and aqueous, and for oil phase, 2.5% consists of spent 80 and white oil. The treatment of this sample using homogenization speed of 600 rpm for 5 minutes and show particle size of 96.03 nanometer and a P of 0.361. Thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.
Okay, thank you, Miss Amalia, for your presentation. And then next, we continue with the third speaker, Miss Winnie Nur Auli. Miss Winnie, time is yours.
Assalamualaikum warahmatullahi wabarakatuh. My name is Winnie Nur Awil. Assalamualaikum warahmatullahi wabarakatuh. My name is Winnie Nur Awili. I will present my paper about in silico character and molecular docking study derivative serum bond for breast cancer therapy. Here, I am from study program of pharmacy at Sumatra Institute of Technology, and we collaborate with Department of Pharmacy Jenderal Sudirman University. The abstract of the paper. So, the objective of this research is to find better potential of breast cancer therapy. The cancer, especially the breast cancer, is already the lead cause of mortality worldwide, and the targeted therapy has been focused to achieve better efficacy and safety. Among all of the natural compound, serum bond from zingiber serum bed or lempuyang has been reported to have anti-cancer activity, but the further research has not been published yet. So this research is aimed to predict derivative of serum bond with the best pharmacokinetic properties and best interaction with breast cancer target. And here we use 33 derivatives of serum bond from many published journals, and then we assess these 33 derivatives. These 33 derivatives, we select the pharmacokinetic properties from Lipinski's rule of 5 using Swiss ADME, and then further we use molecular docking with autodock to predict the interaction with breast cancer target protein, and the binding energy of the interaction were compared with the existing drug with the same mechanism of the protein. After the selection from pharmacokinetic parameter, 21 molecules met Lipinski's rule of 5 and ADME properties, and the 21 selected candidate dock against estrogen receptor and progesterone receptor to evaluate and to get the best derivative serum bond as targeted breast cancer therapy. The binding energy, serum bond derivatives 20 showed lower energy than tamoxifen as the standard of existing drug with the same mechanism with the receptor, and it is showed the potential candidate for further study. So here is the introduction. So the introduction is about the cancer. The breast cancer ranked first in the cancer case with the highest prevalence, but in the other way, the cancer therapy is still in development with many side effects. So there are many many research about the natural compound that has activity of anti-cancer. And about the drug discovery, we find that the conventional method is expensive and time-consuming. So the in silico method approach has been the better solution for drug discovery in the first step. So here, because of that background, we will we are doing the research in silico character and molecular docking study derivative serum bond for breast cancer therapy. And for breast cancer therapy, we use the estrogen receptor and progesterone receptor because among the various types of breast cancer cell subtypes, TNBC or triple negative breast cancer is the most aggressive. And this TNBC fails to express the estrogen and progesterone receptor. Here are the tools that we used, and here is the step of the method. The first one is the pharmacokinetic selection using website Swiss ADME to select the derivative of serum bond based on Lipinski's rule of 5. There are molecular weight hydrogen bond acceptor, hydrogen bond donor, rotatable bond, and also surface area. And then after we select the derivative, we will do the next step, validation of protein. So the estrogen, progesterone, and progesterone receptor were retrieved from PDB website, and each of the protein were validated using the co-crystal natural ligand from PDB to obtain the RMSD below 2. Also, after that, preparation of ligand. The preparation of ligand were built using ChemSketch SCD Lab 2021 and optimized using Gauss view with density functional theory and saved in PDB. After that, we will do the molecular docking. We will calculate the substrate energy Gibbs to and compared with existing drug. After that, we will visualize the interaction about protein and drug or molecule, and we will analyze the binding protein from the molecule and ligand and protein receptor. The result from the pharmacokinetic result, from 33 derivative serum bond, there are 21 molecules that pass the Pinke's rule of 5 as the character of drug molecule. So here the red mark is the are the eliminated derivative of serum bond. And here is the derivative serum bond that we used. We used this derivative serum bond from many published journal, but we prepare or we draw this structure with by ourselves. And then we do the molecular docking. We calculate the binding energy of every derivative serum bond, also natural ligand, and also tamoxifen with estrogen receptor and progesterone receptor. And here the result is that derivative 20 has lower energy than existing drug or tamoxifen. So this is a potential molecule for further study. So here is the visualization using BIOS via discovery study. This is the receptor of estrogen with molecule 20 show the van der Waals alkyl and p-alkyl bond. And also with progesterone receptor, the derivative 20 showed conventional hydrogen bond with the progesterone receptor. For the conclusion, ADMET study and molecular docking study showed that the derivative 20 showed lower energy than existing drug tamoxifen with the same mechanism. Further test of molecule 20 is needed to synthesis. Also, in vitro study and in vivo study in breast cancer cell line specifically. So here we will see again the molecule 20. Here is the molecule 20 that has better or lower energy than tamoxifen. Okay, this is the references that I use for this paper. Thank you for your attention. I will stop this presenting. Thank you.
Okay, thank you very much, Ms. Winnie, for your presentation. And then we will continue for the fourth speaker, Ms. Marisa Angelina. Could you please share your video?
Yeah.
Yes, please. There is no sound. Excuse me.
No sound?
No sound. We cannot hear.
Is there sorry, is there any setting? I hear my sound here. What do you think? Is there any option? How do you think if I done my presentation by myself? Is it okay?
Okay. Do you have any material?
Yes, maybe I will. Oh yes, yes.
Okay.
Wait a minute. Yeah,
Yes.
sorry for the technical problem. Okay, I skip that first. Sorry. How to escape? Why is still no sound?
We cannot hear anything.
Yeah. Wait a minute. Wait a minute, yeah, please.
Okay.
Yes. Okay. Get that. You can see my slide, my voice.
Okay. Yes. So you will present your presentation.
Yes.
Okay.
Is it okay?
Okay. So the time is 10 minutes.
Yes. Okay. Thank you, organizing committee. Please, I'm sorry for the technical problem. I will present our team result with the title LD50 Determination of Caseolata Leaf Ethanol Extract in DIY Mice. I'm Marisa Angelina with my team Indah Didi Wijanti Muhammad Hanafi from Research Center for Pharmaceutical Ingredient and Traditional Medicine BRIN. Yes, we use in this research we use caseolata because caseolata has many biological activity report, and also our team has many tests on caseolata extract. Some of biological activity that report as anti-scabies, anti-malaria, antioxidant, antibacterial, antifungal, and anti-dengue. The compound that report on caseolata are amodine, kilsophanol, kaemphenol, and so on. And for the compound has been reported as antiviral, antibacterial, anti-cancer activity. Why we do the toxicity use the animals? Because from the toxicity use toxicity test use animal, we can see the presence of biochemical, physiological, and pathological reaction that correlated with the toxicity in human. In this research, we done oral acute toxicity test because we want to effect that appear within a short period of time after administration of orally administered test preparation. And LD50 is a single dose of a substance that is statistically expected to kill 50% of experimental animals. The objective of the study is to determine the LD50 from the ethanol extract of caseolata leaf in mice. First, before we do the test, we prepare the sample and do the extraction, use ethanol 70%, and our protocol has been submitted to Ethical Clearance Committee Medical Faculty University of Indonesia. In this test, we use 60 mice, male and female, and we divide into 5 groups. The methods based on the POM non-clinical toxicity test guideline 2014 with our modifications. The mice were acclimatized for 7 days, and also the profile of body weight, behavioral, and neurologic were observed during 14 days. And the mice were fed for 3 until 4 hours before treatment and 1 until 2 hours after treatments. We prepare the sample, use solvent 500 microliter until 1,000 microliter. All mice were given standard feed and drank mineral water ad libitums. The observation during for 14 days, we see the changes on body weight, and also we analyze the blood biochemistry that correlated with liver and kidney function, and also with then histopathology on liver, kidney, and also lung. The result of our test, first, because we use the extract that we prepare before, so we must confirm that the extract is still Cassia alata. Yeah, we can see the profile of Cassia alata. So this test just only used as a quality control of our extract that we've done test before for another bioactivity. After 14 days, we can accumulate the body weight of each sex of mice, female and male, and we can see the increasing of body weight of mice during 14 days is normal. And when we've done the static ANOVA 1-way, there were no significant difference between treatment group and also with the control group. Also, we measure the leukocyte, erythrocyte, hemoglobin, hematocrit, and also we measure the differential count of leukocyte. That we can see there were no significant between control with the treatment group until the dose of 15 gram per kilogram of body weight. From the biochemical profile of liver function, ALT and AST value, we can see the increasing, the most increasing on those 7.5 gram per kilogram of body weight. But when we analyze use ANOVA 1-way, there were no significant with the normal control. Also, we analyze the urea and creatinine value to see the kidney functions. And also, we have a result that there were no significant between treatment group and a normal group. Yes, we've done the histology. This is the histological profile of kidney. We can see the tabuli of kidney. In this histological profile of liver tissue, we can see the perivascular infiltrations, and also we analyze the central vein and the lungs of mice. We can see the perivascular interstitials. In the study, 14 days after administration of the extract, there were no deaths for level of dose and also a control normal. So it can be concluded that extract had LD50 more than 15 gram per kilogram of body weight. The result of all hematological parameters showed not significant difference with a normal control, P more than 0.05, and were within the internationally accepted reference range for its test parameter. It could be suggested that the ethanol leaf extract of Cassia alata did not produce adverse effect on the bone marrow. The result also implied that Cassia alata may not have negative effect to the immune system since the white blood cell and differential count are not impaired. And also the result on routine blood profile as well as blood biochemical measurement related with liver function and kidney function showed no detrimental effect on kidney function of mice and cannot cause an impairment of liver function of mice. And also histological observation of kidney, liver, and lung tissue showed that there appear to be tubular degeneration is both control group, normal up to a dose 15 gram per kilogram body weight, and to be a slight inflammatory reaction in the test group. However, this is an adaptive reaction of the cells to the foreign substance and its reversible effect. There was vascular degeneration and perivascular inflammatory infiltrate in the normal control group up to a dose 15 gram per kilogram of body weight, and fatty change and perivascular inflammatory infiltrate are acute reactions that are reversible effect and occur because of the defense mechanism of cell against foreign substance. Through the adaptation process, they will return to a normal condition. The conclusion of our test, the LD50 from Cassia alata ethanol extract more than 15 gram per kilogram body weight and group into practically non-toxic substance. Thank you for the time. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much for your presentation, Miss Marisa. So, four speakers already present their presentation. Thank you once again for all presenters for your inspiring presentation. And then now we entering the discussion.
We cannot hear your sound.
Hello, sorry. I'm sorry. Okay. Is there any question for the audience? Okay. So maybe I have a question for Miss Ivana. Are you there, Miss Ivana?
Yes, I'm here.
Yeah. So I have a question. Could you please explain more detail about the preparation of the plant samples before the extraction procedure?
Okay. So we collect the plant from Simelu Island in 2017. We collect the leaves, stem, and fruits, and the part of plant we separated and washed, and then dry under sunlight. And the dried plant amount of 10 gram we crush into a fine powder and keep at 4 degrees Celsius for further use. And the plant powder was extracted for 3 days with N-hexane, dichloromethane, ethyl acetate, and methanol. And then the filtrate was collected and concentrated using a rotatory evaporator at 35 degrees Celsius to get the crude extract. And the extract was like, is it clearly enough?
You dry your sample with sunshine?
Yes. Yeah, under
Okay.
the sunlight, yes.
Okay. Okay, thank you very much, Miss Ivana. So anyone?
Yes,
Do you have any question?
I want to ask the second
Okay.
speaker, but
Please.
I cannot remember the name. Could you please present
The
about ringan nano emulsion?
Yes, Miss Amalia, the
Miss
second
Amalia.
speaker.
Yes, Miss
Okay.
Amalia, there's interesting research, yeah. But I want to know, I can, from the picture of your presentation, I can see that the emulsion in white color. That we know your extract is black, maybe, yeah, black or browns like an usual extract. How could the nano emulsion didn't affect by the color of extract? Is it the concentration of your moringa very low? Could you explain?
Yes, thank you for your question. Why didn't affect the nano emulsion? Because yes, the concentration of extract moringa is very low.
May I know the concentration on the extract on your nano emulsion?
It's just for below 5.
5 what? The
5%.
Oh, also, sorry, it is discussion, yeah, not testing your result. But also, I have experience on make emulsion on concentrate 2.5% of the extract. The emulsion still coloring. So the emulsion like browns coloring. How could your emotion still white? Is it really interesting for me? Maybe you can see 5%, stay below 5% in total.
Maybe because the base water is large.
The base water is large.
Yeah. Because the base water is used 97.5%.
Yeah, it's also happened to me. But your extract is in brown coloring.
If the concentration is high, there are, but not really brown.
But your extract before you give to the base is the color is brown.
Yeah.
Okay. Yeah. Thank you, Miss Amalia. Success
Yeah.
on your research.
Okay.
Thank you,
Miss Marisa.
moderators.
Okay, thank you, Miss Marisa, for your comments. Any other question for the audience, from the audience? Okay. Okay, maybe I have a question for Miss Winnie, the third speaker. Are you there, Miss Winnie? Miss Winnie? Okay, I think there is a poor connection of Miss Winnie. So, is there any question for the
Maybe I can ask the one question to Miss Evana.
Okay, please. Yes.
Miss Avana, your research is very interesting. But I want to know is there correlation between antioxidant and antibacterial? Because I have many presentation today and also yesterday. There's many antioxidant and antibacterial testing on the extract or sample. Is there any correlation based on your experience, based on your maybe study? Thank you.
Okay, thank you, Miss Marisa, for your question. I think there is a relation about antibacterial and antioxidant activity from my result. It showed that from the result, the more polar solvent attract more chemical compound with antibacterial and antioxidant property. Based on our result, methanol is more efficient attract the antibacterial and the antioxidant compound. Maybe from the component, maybe from the component of the extract, from the methanol Ficus septicalypse, the component is total phenolic, and also maybe I think in the antibacterial activity, the same compound, because because the same solvent have the greatest antibacterial or antioxidant activity. It's from methanol.
Okay, yeah. Maybe you can make some study like correlation study. Yeah, I don't think so, but if I'm not mistaken, there's statistically method to make the correlation, so you can get a new paper.
Yeah. Okay.
Thank you, Miss Ivana.
Yeah, you're welcome, Miss Marisa.
Okay, thank you very much. Is there any other question from the audience? Okay, I have a question for Miss Marisa.
Yes.
Yes. According to your result, the extract contain non-toxic substances, right?
Yeah.
Yeah. So, in your opinion, what kind of dosage form you suggest for this extract to be formulated for the further study?
Yes. This test only report the acute effect. So until the 15 gram per kilogram of body weight, there were no days. So the group to practically non-toxic is formulae from the book of toxicity. If you get the LD50 more than 50 gram, it means your substance practically non-toxic. When we want to suggest the dose, we must do some research. We must do the efficacy on maybe we claim it's antiviral, so we must do the efficacy test on the antiviral. The name of test pharmacodynamic, and then we must continue our test into a subchronic toxicity test. Based on that, maybe we can suggest the best formula or the safe formula to administer maybe for the human or also for the mice as a testing animal.
Okay. Good luck, Miss Marisa. Thank you
Thank
very
you,
much.
Sandra.
Yes. Okay, any other question from the audience? Okay. Maybe I have a question for Miss Winnie. Miss Winnie, are you there?
Yes, I'm
Okay.
here.
Yes. Your presentation is very interesting. So I have one question. How did you validate your method to make sure the in silico screening method that you use in this study was already established and give reliable result?
Okay, thank you for the question. So I validate the method using the co-crystal ligand, co-crystal protein from the PDB or protein data bank. So from the protein data bank, we have the protein and also we have the natural ligand of the protein, and then we dock. And then we separate the natural ligand and also the protein, and then we redock the separate natural ligand and also the protein using the autodock tools. And then after the autodock tools have reached the RMSD as the validated parameter below 2, so the method already validated and can be used for the sample or for the derivative of the serum bond. Thank you.
Thank you very much. And then one more question. Is it okay? Yeah.
Okay.
Okay. So, you have found a compound number 20 as potential candidate, right? with the
Yes.
lower binding energy
Yes.
than tamoxifen. So
The existing drug. Okay.
yes, based on your opinion, which part of compound 20 structure or functional group
Okay.
of this compound responsible for its activity compared to serum bond or other derivatives?
Okay, so the most potential is because of the hydrogen bond of the structure. So hydrogen bond, so the hydrogen bond from the structure is the main or the the most important bond for this interaction. That is the bond. So for the structure, any structure from the structure of the serum bond that can be used from hydrogen bond, all of the functional group is available or very or it is can be potential if they have a hydrogen bond, such as a carboxylate group or such as amine group.
Okay, thank you very much for your explanation, Miss Winnie. Okay, for the audience, any other question or comments? You can write down on the chat room. Okay. So maybe I have one more question for Miss Amalia. Are you there, Miss Amalia?
Yeah.
Yes. So thank you very much for your presentation. It's very interesting. So I just want to know about your presentation, especially on slide 6. I want to know what is the difference among its samples you study. So you have several samples to be tested for nano emulsion, right?
Yeah.
Yes. So you just write down the code of samples on the table.
Yeah.
So could you please explain what's the difference among each samples?
The difference is from percentage of base water.
Oh, the percentage of base water.
Yes.
Oh, okay. And then from that table, you have already explained that the lower PI value means better homogeneous particle size in that sample. According to the result, there are several samples with lower PI value than sample K. So, and why did you conclude that sample K was the most optimum nanoemulsion?
Because I see in the particle size.
So, based on its particle size.
Yes.
Oh, okay. So the PI value is like another consideration.
Yeah.
Oh, okay. Thank you very much, Miss Amalia.
Okay. Thank you, Miss.
Okay, for all audience or participants, is there any other questions or comments? Okay. Okay, if there is no questions and comments anymore, we will close this session. For all presenters and audience, thank you very much for your kind and very active participation during this session. After this, please return to the main room to join the plenary session, which will be started at about 3 p.m. Okay, so once again, thank you very much and have a nice day. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Kamila.
Welcome,
Waalaikumsalam
everyone.
warahmatullahi wabarakatuh.
Recording in progress.
Would you like me to already start sharing my screen? Would you like me to start sharing my screen already?
Not yet, sir. Or you want to try the technical matters right now?
Yeah, but I think, I can just try for a second.
Yeah, you can try before we start again at 3.
Just a second.
Yes.
Yeah.
Let's see.
Yes, we can see your slide clearly.
Yeah, that's it. That's fine.
Yes.
Okay, I'll stop sharing now then.
It's
and
okay.
you can put it on
you.
again. And let me see. Stop sharing. Yes.
Okay.
Good.
Thank you, Dr. Arnold. Menit lagi. Ladies and gentlemen, so happy to welcome you back on the main room. So the next agenda is plenary sessions. Therefore, may I invite Dr. Hayati Iskandar as the moderator of the sessions. But before that, I would like to read short profile of her. Dr. Hayati Minarsi Iskandar is a researcher of Indonesian Oil Palm Research Institute who are interested in molecular biology, biotechnology, and biochemistry. After graduated from Bogor Agricultural University or IPB, she continued her master degree in plant molecular biology at Vrije Universiteit Brussels, Belgium, and obtained her PhD degree in biochemistry and molecular biology at the University of Queensland, Australia. Without further ado, please welcome Dr. Hayati Iskandar. Dr. Hayati Iskandar, time is yours to lead the sessions.
Okay. Thank you, Ibu Aulia, for my introduction. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon in Indonesian time, distinguished guests, all the speakers, and all participants. Welcome to the next plenary session of the 9th International Symposium of Innovative Bioproduction Indonesia on Biotechnology and Bioengineering, or ISIBio 9 2022. Please allow me, Hayati M. Iskandar from IOPRI Bogor, as a moderator to lead this session. This afternoon, initially, we will have two guest speakers, Dr. Arnaud Bovy from the Netherlands and Professor Chin Kun Wang from Taiwan. Unfortunately, Professor Wang is not able to attend this session because he suddenly has to conduct a surgery in a hospital. So we will start the plenary session with the presentation of Dr. Arnaud Bovy. Am I right to pronounce your name, Dr. Arnaud?
Yes, you're right.
Okay,
Absolutely.
thank you. First, I would like to introduce you about Dr. Arnaud Bovy. So his CV. Dr. Arnaud Bovy, he's a researcher at Department of Plant Breeding, Wageningen University and Research. He's also a group leader at Breeding for Quality Research Group, also at Wageningen University. And his research interest is metabolomics, plant breeding, and genetics. So Dr. Arnaud will present an interesting topic about application of genetics and metabolomic in assisting plant breeding for quality. So you will have 20 minutes for presentation, and we will have a Q&A session after that. So Dr. Arnaud, the time is yours. Thank you.
Thank you very much. I will now share my screen. Okay, well, good afternoon, everybody. It's a real honor for me to be invited to give a talk at this conference. I really would like to thank Wayuni and the other organizers for inviting me here. And well, the title of my talk in today's session is Application of Genetics and Metabolomics in Assisting Plant Breeding for Quality. And so, as was already mentioned in the announcement, I lead a research group on breeding for quality within the plant breeding department of Wageningen University and Research. And in my talk today, I will talk about the combination of genetics and metabolomics in order to assist breeding for quality. So we are in the plant breeding department. And well, actually, I would like to start with the question, what breeders want? What do they want in their breeding? First of all, they would like always to find novel sources of their traits. And in particular, they are looking for genetic variation, and in particular, crossable genetic variation with which they can, let's say, improve their crop with traits of interest. Breeders also would like to have tools to select for their traits. And on the one hand, these are efficient phenotyping tools, but more and more important in breeding is to have genotyping tools so that breeders can already select for a trait of interest at seedling stage by using molecular markers. And important for this story is that the best possible molecular marker actually is the gene that's underlying the trait of interest. In addition to breeding for yield and disease resistance, there's a growing interest to also breed for quality traits, and that's the topic of my work. And if you look at these quality traits, you can basically look at or see a number of traits that you can visually monitor, such as size, shape, color, and shelf life of your crop. But there are also invisible traits of interest, like nutritional value, taste and aroma, and pungency. And especially in order to monitor these invisible traits, we need a good detection platform. And many of these quality traits are determined by small molecules that are present in our fruits and vegetables, small metabolites, and detecting these metabolites is one of the main aims of our research. Well, we do that using metabolomics, and metabolomics is a technology aimed to provide a broad overview of the metabolic composition of a given extract. And metabolomics, as we use it, is based on mass spectrometry, which basically stems from the idea that every molecule has its own specific mass and can be detected as such. Well, what are the advantages of metabolomics for breeding? First of all, metabolomics can give you the insight in the metabolic basis of often complex traits. It also allows you to define subtraits. And defining subtraits is a way to simplify genetics, as I will show you in a minute. And also metabolomics and knowledge of pathways helps you in identifying genes that may be involved in this process. And to illustrate that, I would like to give you an example of a complex trait, taste, a trait that we worked on a lot in particular in tomato. And when you want to study the genetics of taste, the first level you can do that is by measuring taste appreciation using consumer panels. But you can imagine that just giving an evaluation of liking is not giving you a lot of differentiation in the traits. So a second level at which you can look at taste is at the level of trained tasting panels. And these trained tasting panels, they are able to discriminate many more aspects of taste. For example, they can dissect taste into, for example, taste, aftertaste, mouthfeel, and aroma attributes. And under these attributes, there are different sub-traits like sweetness, sourness, umami, and so forth. This is already a new level of detail. But if we would look at the metabolites underlying these traits, we can see that under each of these attributes, there are multiple players that contribute, multiple metabolites that contribute to this particular trait. So in fact, this very complex trait, taste, can be divided in many different subtraits by looking at the metabolites. And looking at these metabolites, studying these metabolites, also gives you hints on the biochemical pathways that are important and the genes that may be involved in different steps of these pathways. So that's a big advantage of studying metabolomics. Well, in addition to metabolomics, today's Breeders' Toolbox has really expanded. And it has expanded, for example, by the availability of genome sequences. And if you look at a crop like pepper, more than 400 accessions have already been sequenced today. In tomato, it's even more than 700 at the moment. There are high throughput marker systems available to genotype large numbers of genotypes. Transcriptomic tools and databases are available, and of course the metabolomics platforms. In addition to these tools, also there are more and more interesting breeding materials available. For example, the presence of mapping populations that you can use to study the genetics of your trait. A specific type of mapping population that I'm going to talk about today is a GWAS panel, which basically is a diversity panel of accessions. Tilling populations are available nowadays, so mutagenized populations that you can use to identify mutations in specific genes of interest. And of course, transformation technology, which is available, very important to, for example, study genes by virus-induced gene silencing, or make stable knockouts using CRISPR-Cas gene editing. Well, in my talk today, the high-throughput marker platforms, metabolomics, and the use of GWAS panels is going to play an important role in the example I would like to give. It may be good to mention, in my research, tomato and pepper are the major crops that we work on, and the talk today, as you can see from the pictures, will be about pepper. The topic of my talk today was studied in a big EU project called G2P-SOL, Genotype to Phenotype SOL. It is a large EU project that just ended at the end of last year. And the aim of this big program was to link genetic resources, genomes and phenotypes of solanaceous crops, tomato, pepper, potato, and eggplant. And my involvement was mainly in the pepper part of this work. The aims of this project was to do an inventory and conservation of material stored in gene banks. And it was mainly done by high-throughput genotyping. I will come back to that in a minute. A description, assessment, and phenotyping of the collection, genetic analysis of this phenotypic variation, and there was a pre-breeding part in this program. Now, I already mentioned that our main involvement was in pepper, and as I already mentioned, also genetic diversity that was studied is the diversity stored in gene banks. And for each of the crops, more than 10,000 different accessions were genotyped using genotyping by sequencing or other next NGS-based platforms. So for capsicum, 10,000 different capsicum accessions were sequenced. Most of them were capsicum annuum accessions, but also a number of accessions from Bacatum, Chinense, and Frutescens. And let's say the genotyping and the insights we gained about the evolutionary history of pepper has been published in this nice paper of Pascale Tripodi in PNAS last year. Well, based on the genotypic analysis and the genotypic diversity that was found, a core collection was made consisting of 450 capsicum annuum accessions. And this core collection was studied further as part of the project. And because this core collection was derived from the genotypic data, it was expected to represent the genetic diversity within capsicum annuum. And this core collection was further analyzed by high-density genotyping and by phenotyping of 3 trials conducted in different countries in Europe. Well, here you just get a small overview of the phenotypic variation that was present in this collection. So there's a huge amount of variation. Now, what we tried to do in the past year was to get a better understanding of the metabolic diversity in this pepper core collection. And if we think about the metabolic composition of pepper fruit, actually pepper makes a lot of very interesting compounds. Compounds involved in color formation, like carotenoids, chlorophyll, and anthocyanins. Compounds involved in providing pepper with taste and aroma, like volatiles, sugars, acids, and capsaicinoids. And other nutrients, dietary nutrients, like vitamins, capsaicinoids, and capsiates. So we were aiming to measure these compounds in this pepper core collection. Now, the trial that we conducted looked as follows. The collection consists about 400 capsicum annuum accessions, 20 Chinenses, Frutescens, and Bacatum collections. They were grown at different locations, and I will talk today about material grown in France and grown in Italy. And ripe fruits of these trials were harvested and subjected to different metabolomics analysis. So basically, LCMS to detect semipolar secondary metabolites, and GCMS to detect volatile metabolites. Now, how many compounds can we detect using these platforms? Now, if we look at the GCMS platform that detects volatiles, we detected 128 different volatiles in the INRA trial in France and 133 metabolites in the trial in Italy. And most of the compounds that we detect, we can actually identify. And for the GCMS, we only had, let's say, 10 unknowns in total. With LCMS, it's a completely different story. In LCMS, we were able to detect, let's say, hundreds of compounds, but the majority of these compounds actually are unknown, and identification of compounds using LCMS is still a big challenge in metabolomics research. And the groups that we could detect consisted of capsaicinoids, flavonoids and other phenolics, and capsicanoids, which are diterpenoid compounds. Now, if we look at the distribution of these metabolites over the different accessions, you can visualize that using a principal components analysis. And in this principal components analysis you see here, actually all the dots represent the different accessions in our trial, and with the arrows, the main drivers of the variation are shown. And you can see a clear division in the first axis of the pungent genotypes on the right side and the non-pungent capsicum onion peppers on the left side. And if we look at different, let's say, volatile compounds and taste-related compounds, we can see that on the right corner here is the place where most of the wild accessions end up, and they are, let's say, the richest in volatile esters, which usually give a fruity aroma to your fruit. So this gives you some insight in the metabolic variation, in this case for volatiles in this material. Now, there are hundreds of compounds that we can detect, but which are the ones to focus on? Which are the ones that are important, for example, for taste? Well, one way to do that is to see if you can find correlations between the presence of certain compounds and a certain taste attribute. And using a few colleagues, we basically determined the aroma and the taste of all these peppers. And we could discriminate, for example, 3 different types of aroma. The beany green pepper aroma that you find mostly in blocky peppers, a more grassy green fleshy cut grass aroma, and a sweet fruity aroma. And when we look at the correlations of these taste attributes with different metabolites, we can clearly see that methoxypyrazine is the compound that really is responsible for this beany green typical blocky pepper aroma. If we look at the grassy green type of aroma, it's mostly the lipid-derived volatiles that are highly correlated with this type aroma, and that is very well in accordance with also taste in tomato, where these compounds also play a very important role. And if you look at the sweet fruity aroma, it's especially a number of volatile esters that are highly correlated with sweet fruity aroma. So in this way, we can basically select a number of potential target compounds that we could use in breeding for taste. And for example, if you would like to breed for a pepper with a more sweet and fruity aroma, we would aim at increasing the amount of esters and decrease the amount of these methoxypyrazine or oxylipin-derived volatiles. So these could be potential breeding targets for taste in pepper. Well, we also tried to see if we could link the metabolic variation that we see to genetic variation. And to do that, we used a GWAS study, and GWAS stands for Genome-Wide Association Study. So basically what we try to do using molecular markers is to find associations between specific molecular markers and specific compounds. So the data we used for this genome-wide association study were these volatile aroma compounds and the secondary non-volatile metabolites, around 200 metabolites in total. We used the data from the 2 trials, from the 2 different locations, and we used SNP data from genotyping by sequencing, and we used in total 10,000 SNPs in this analysis. Now, we used also 2 different, let's say, standard approaches that are used in GWAS, mixed linear models and a blink approach. Now, in the GWAS, we could find a number of common QTLs that were found in both location trials. In total, we found QTLs for 105 metabolites at 524 different loci. And the first one I'd like to show you here is the GWAS analysis. GWAS results often are visualized in this kind of what we call Manhattan plots that here show you the distribution of the 12 chromosomes of pepper, and here the association of your compound with specific loci on the different chromosomes. And you can see here a very nice association at the end of chromosome 2 of capsaicin, the compound responsible for pungent taste in pepper, and this location on chromosome 2. And it's known that this location is the famous PUN1 gene, which is the gene that's the ester-forming enzyme responsible for the last step in the biosynthesis of capsaicin. And this very nicely matches with earlier results in which gene bank annotations for pungency also showed up a very nice QTL at exactly the same spot. Another observation that we did, and relevant for this story, is the fact that biosynthetically similar compounds, they correlate quantitatively with each other. And to show you that, we made this, let's say, this hierarchical cluster here, and each of the colors actually represents metabolites that are closely related to each other. And you can see that these metabolites form rather separate clusters. So it means that metabolites with the same origin or the same biosynthetic pathway are often correlated with each other, which would suggest that the regulation of these pathways may be regulated by hotspot QTLs. And the question is, do we see this kind of hotspot QTLs? And actually, when you look at this graph, here, this is actually a merger of all the QTL data that we found in our study. And we tried to visualize these data using bubble sizes. And the larger a bubble size, the more compounds are actually co-localized on the same QTL. So, for example, if you look at the bubble size here, at the position of the QTL for capsaicin, actually, we have here not only one different capsaicin, but a number of capsaicins that show the QTL at this particular position. And that is because this ester-forming enzyme, PUN1, does not only use one specific substrate, but has a broader specificity for different substrates, leading to different capsaicinoids. The same type of very nice bubble here, we see a QTL for different flavonoids. And we see this QTL at the end of chromosome 5. And our most associated marker was landing exactly in the MIP12-like transcription factor. And MIP12-like transcription factors are actually known to regulate the flavonoid pathway. And we cloned the MIP12-like gene from tomato a number of years ago and showed that these transcription factors indeed are regulating the flavonoid pathway. Well, actually in pepper, we find a very nice candidate gene called MIP12-like, which seems to regulate also the flavonoid pathway in pepper. The MIP12 gene in tomato is actually located on chromosome 1, and this ortholog is located on chromosome 5 of pepper. So we found quite a number of nice and interesting QTLs. But one of the main challenges of GWAS studies is to validate these QTLs, to make sure that they are really showing you the right lead and not false positive, for example. And one of the QTLs we validated was actually this MIP12-like QTL I was just talking about. And it was very nice to validate this QTL because we started that work already quite some years ago. And it was actually the work of Wayuni, and here you see a picture of Wayuni 10 years ago when she was still working in Wageningen, and she identified 2 accessions We call them here for simplicity P12 and P24. P12 was a very high flavonoid accumulating accession, and P24 was a very high vitamin E accumulating accession, but low in flavonoids. And in collaboration with Ilan Paran in the Volcani Institute of Israel, we developed segregating populations based on the cross of these two parents. And when you make segregating populations in the F2, these nicely segregate, and every F2 individual basically is a genetically unique individual. So we did a QTL analysis of this F2 population, and actually in this population, we also find a very strong QTL at the same position on chromosome 5. This QTL, you can see here that if you have the P12 allele of this QTL, you get much higher flavonoid levels than when you have the P24 QTL allele. And also what we found was that a large number of flavonoid genes showed a much higher expression when the P12 allele is present compared to when the P24 allele is present. And this, of course, suggests that there is a transcription factor into play here. And actually that also led us, independently of the GWAS trial, to the identification of the MIP12-like gene. Now, we did a number of experiments to indeed validate and show that this MIP12 gene is indeed the gene doing the job. One of the things we did is we knocked out the MIP12-like gene in this parent 12, the flavonoid accumulating parent, using virus-induced gene silencing. And you can see if you have the flavonoid level in the control, here plants that we in which we used a GUS construct to silence, versus the MIP12-like silencing, you can see a clear decrease in flavonoid content in the silenced plants. We also try to overexpress the gene in a pink tomato background, so in a MIP12 mutant of tomato. And also here you see that when you have high expressors of the MIP12-like gene from pepper, you can see a clear increase in flavonoid content compared to the low expressors or the controls. And last but not least, very important for breeders, is can we use this gene actually to increase the flavonoid content of commercial materials. And we did that in a few backgrounds, and I'll show you here the results of introgression in sweet banana. And you can see when you introgress the P12 allele, you really get a nice increase in the flavonoid content in sweet banana compared to, let's say, the control without this allele. So also for breeding purposes, this gene is a very nice marker, and P12 is a very nice donor for this trait. Now, the ongoing research is actually focused on validating QTLs that we found in the GWAS in biparental segregating populations. We are mining these QTL regions for candidate genes and hope to identify and analyze candidate genes. We also want to improve our GWAS study because our study was really limited in the number of markers. I already mentioned we use 10,000 markers, but this is far too low for a GWAS study. And that means that we are certainly still missing maybe important QTLs because we don't detect the right alleles. We are trying to alleviate this problem by sequencing the entire core collection, and this is something that will be done in the near future. And finally, we'd like to also combine different data types. For example, we would like to combine our metabolites with taste, as I showed you, but also with, for example, shelf life data. Now, finally, I would like to come to the conclusions. I think that gene banks harbor a very wonderful resource of genetic variation that can be exploited by both researchers and breeders. The breeder's toolbox has really expanded in recent years with various exomics platforms, making it possible to have a much more detailed look and getting detailed insight in our traits. I hope I showed you that metabolomics is very useful to get insight in the metabolic variation in a crop, in the metabolic basis of traits, and in the genetic basis of traits. And finally, GWAS provides novel donors, QTLs and markers for traits of interest. But an important part, and that will take years to come, is the validation of the QTLs that we found. So last but not least, I would like to acknowledge the people involved in this research. From my own team in Wageningen, Yuri Tikhonov, he plays an important role in all the research we do. Jos Molthoven and Fien Meijer, two technicians in the project. Yi Wu, a PhD student who participated in this work. And then Richard Vinkers, Matthijs Brouwer, and Giorgio Tumino, who were involved in the statistical analysis of the GWAS data. We collaborated with people in France and in CREA to set up these GWAS trials, and with Ilan Paran from the Volcani Center, in which we collaborate closely in this research. And finally, all the partners that participated in G2P-Sol, who made it possible to have such a large-scale project going on successfully. And this is where I'd like to end my presentation, and I'm happy to answer some questions. Thank you.
Thank you, Dr. Arno, for the nice presentation. So, ladies and gentlemen, now we will have a question and answer session for about maybe 10 minutes. So you can deliver your question by chat box or raise hand. So please, you can start to give questions to Dr. Arno. Okay, now I have one from the chat box.
Yeah, very nice question indeed. Okay. For a human tasting panel, won't capsaicin ruin your taste bud to differentiate other tastes and aromas? Yeah, it's a very good question, and especially for Western people, it's of course very difficult to taste anything once you taste capsaicins. So if you talk about aromas, it's not so problematic because you don't taste it, but you just smell. But for really the combination of tasting and smelling, we cannot do that ourselves. And we did that in the past using actually Indonesian people to do the tasting, because they can much better differentiate tastes in the background of high pungency.
Okay, thank you, Pak Dani Satyawan from BRIN. Is there answer your question?
Yes, and it's quite amusing actually to find that some Indonesians actually have some use in this kind of studies. Thank you, it's very amusing.
Okay, thank you. Next, please, for the next questions. Or maybe I can also ask you, Dr. Arno.
Yes.
I'm interested when you said that metabolomics or metabolic is basis usually used for the complex traits. So what about the, like disease tolerant or resistant? Yeah, because we know in Indonesia that are the big problems, especially for this chili pepper. You know, Indonesia very love chili pepper, chili fruit or chili pepper very well. So maybe you can explain about this. Can we use also this metabolics?
No, certainly this can be done, and we have very nice examples of it also. So also in the basis of resistance, metabolites play a role. And indeed, metabolomics can be used to get a better insight in the mechanisms that the plant uses for resistance against pathogens. And especially also when you want to, for example, get insight in the type of genes involved, a very detailed insight in the structures and the metabolites that are playing a role there, it can really help in identifying the underlying genes.
Okay.
Yes, it's definitely a very nice combination to combine resistance and metabolomics.
Okay, thank you, Dr. Arno. I see that Dr. Wahyuni is raising hand. Please, Dr. Wahyuni.
Yeah, thank you, Bu Hayati.
Yeah.
Hi, Arnold. Thank you for including my pictures in your slide.
Well,
Well,
you made the basis for our paperwork, as you know. So
yeah, I always with your G2P Sol project. It's a very big project, including at least 400 capsicum annuum accessions, in addition to Pakatum, Chinensa, and other species.
yes.
And I saw the PCA of the volatile compounds. It's ended similar. I mean, the volatile compounds with this big hundreds of accession, they differ the accession based on the pungency level. Like we have in the last time that we used 32 accessions. Yeah. And then with this project, have you already identified if whether these volatiles came from or derived from the capsaicinoid pathway?
Yeah.
And yeah, the second
Yeah,
question, Arnold, or later.
Shall I first answer your question? Your first question?
Yes, please, Arnold.
Because you know this is a very interesting question, because what we indeed see is that pungent accessions make a lot of volatiles, specific volatiles that you only find in the pungent accessions, and we don't know if this is, let's say, due to a really a genetic, for example, the PUN1 locus, whether that is also involved in producing specific volatiles, because it's an ester. PUN1 is an ester-forming enzyme, so basically you could expect that it can also, for example, produce volatiles, or whether this is due to the fact that it's just an association, because these are two different sets of germplasm, the hot germplasm versus the non-pungent germplasm. Actually, we still don't have the real answer to that question, Juni. We had the question already in the time that you were working in Wageningen, but it's still an open question. And but we know what type of experiments to do to show it.
Okay.
So we really need a PUN1 to make a PUN1 mutant, an isogenic PUN1 mutant that in a pungent background, and that I think will enable us to answer that question.
Okay, so this one of the complex traits, right? These volatiles.
Yeah, sure.
Yeah.
Yes.
And
no, so we are very interested in answering that question, but didn't do the right experiments yet to answer it.
Yeah. That's one question that I want to ask you. If we have this complex traits, and can you suggest, or well, at least if you have something in mind, what kind of experiment that we should design to answer some kind of, for example, the markers underlying these complex traits?
Yeah, I think the problem with complex traits is that genetic studies are difficult because the complexity is too large, and you don't have the statistical power to detect all the players that contribute to your complex trait. So that's why we think it's important to really define subtraits, which we do based on, for example, metabolites, and then study each of these subtraits, because it will make genetics simpler. So that's one thing I would advise to do, because if you only look at the phenotype of the complex traits, probably you're not going to get there. So getting insight in your trait is very important, and so metabolomics is one way to do that.
Yeah,
is that answering your question?
Yeah, well, yeah, metabolomics.
And then of course, the next step is when you are able to identify, for example, markers for different of these subtraits, of course, you have to prioritize them based on the contribution that they make to the final outcome of your complex trait. And that is something you have to evaluate, for example, from your QTL analysis to see what is the percentage explained variation for each of these different subtraits. And then you can focus on the ones that are giving you the best possible outcome.
Yeah. Okay, Arnold, thank you.
Okay, thank you, Bu Wahyuni. So we still have now from the chat box from Bapak Dylan Lowry.
Let's see.
How would you plan to approach the unknown compound that detected in your LC and GCMS analysis?
Yeah. So as I already mentioned, in GCMS, it's not such a big problem. We have only a few unknown compounds, but in LCMS, that's really a problem. Now, we don't aim to identify all these compounds. That sometimes it can take a PhD study to identify a few compounds. It can be very difficult. That's why what we do is we use these unknowns in our studies. But only when we can see that an unknown compound really contributes to a trait of interest, it is worth for us the effort to try to identify it. And the way to identify it, for example, is to do a much more elaborate LCMS study with MS fragmentation in order to really find out what the structure of your compound is, just as an example. But our approach is very on purpose. Don't bother too much about the identification in the beginning, but do that at the end when you know that these compounds are really important.
Okay. Mr. Dylan.
That was, I think, the answer to the question. Yeah.
Okay.
Then how would you know that the unknown is gonna contribute significantly to the traits?
Okay, so well, one way to know that is to do a correlation analysis to see if there's a correlation between the presence of your compound and your trait of interest, as I showed you for example, for taste. And when you have a certain taste attribute and you look at the correlations of all your metabolites with this particular attribute, that's one way to, for example, look at this correlation. But there are many different ways to do that. One other way that for example, we like very much at the moment is when you are interested in taste and you run a GCMS analysis, one of the things you could do is to connect your GCMS device to an olfactory port and smell each of the compounds that comes off your GC column. And in that way, you can also detect specific flavors and detect specific compounds. And what we have seen is that sometimes compounds that are highly detectable and give you very small signals can give a very powerful taste experience, and also the other way around. So this is another way to find out, okay, I don't know the compound, but this one we need to focus on.
Okay, Dr. Arnold, we still have one more question, and maybe this is the last question for this session from Dr. Penny Lestari, or Ibu Penny Lestari. So, on the level of pungency of chili is also influenced by the environment, such as dry and other stress. Okay.
Yeah, this is an interesting question. It is known indeed that capsaicins are strongly influenced by the environment, and I know that breeders are very interested in ensuring that they have a stable amount of capsaicins in their crop that's less influenced by the environment. But we don't, I at least don't have the knowledge whether how big that influence is and whether this indeed changes with shelf life. It's a good question, but I don't have the answer.
Okay, so Bu Penny, maybe we can further discuss with Dr. Arno after this session. Okay, So thank you for the fruitful discussion. Oh, there's one more. Maybe Dr. Arno can answer with a brief question. These questions in briefly.
So which is the most reliable result between genetics and metabolomics? I don't know exactly what you mean with this question, because I think it depends reliable for what. So that's hard to answer. But both platforms have their pros and cons, and I think that especially the combination is strong. If we, for example, see that compounds that are correlating also end up at the same regions, also genetically show the same QTLs and so on, well, this gives us confidence, for example, that a certain compound may be involved in a certain trait.
Okay, thank you, Ibu
Yeah,
Rita.
so it's basically using the correlation on the one hand, genetics on the other hand. If everything points in the same direction, that gives you a certain level of reliability.
Yeah, but metabolomics also needs the genetics information, right?
Yeah, you know, when you want to do metabolomics and you would like to look at variation, or you need variation in order to see differences, and our approach is always based on detecting differences. And there, you know, having, for example, genetically diverse material is a must. Another way to create diversity, of course, is different treatments. We have focused much on using genetics, but you can also use treatments. But especially also in relation to the question of the identification of compounds, when you don't have variation, it's also impossible to, for example, prioritize compounds to focus on. So that, I think, discriminates also metabolomics from, let's say, the more classical biochemical studies that are really aimed at identifying every compound. But we are really focusing on the ones that do the job, that are important for our traits.
Okay. Thank you for the questions. So, and also this fruitful discussion for Dr. Arno. So maybe in the future, if we can see from your presentation, this metabolomics and GWAS or genome-wide association study will be more improved, and many people maybe use it for in breeding session, right? Okay. So again, we appreciate Dr. Arno Bovi for the excellent presentation. Maybe by giving him a big applause. Thank you for your attention also, everyone, and I will close this session. For the next program, I'll return to the committee or the Master of Ceremony. Thank you again, Dr. Arno.
Thank you too. It was a pleasure.
Thank you.
Thank you, Dr. Arnold, for the interesting materials. And also thank you to Dr. Hayati for leading the sessions this afternoon. Very fresh, ladies and gentlemen. So for the next agenda, we are going to announce best oral presentations. Who is the best oral presentations during 2 days of ISIBio? Any guess, ladies and gentlemen? And the best oral presentations goes to, drum rolls, any guess? Yes, congratulations to Dr. Sugiono Saputra from Research Center for Applied Zoology with the title of Sago's chart hydrosate as a carbon source for expolysaccharide productions by Lactobacillus plantarum. Congratulations for Dr. Sugiono as the best presenter at the NIN International Symposium of Innovative Bioproductions for Biotechnology and Bioengineering, ISIBio. Please give a round of applause, everyone. Ladies and gentlemen, and now I would like to invite Mr. Ahmad Fatoni, PhD, Head of Research Center for Applied Microbiology, National Research and Innovation Agency, to convey closing remarks. Bapak, screen is yours. Still unmute.
Yeah, sorry. Yeah. Sorry. Yeah, thank you, Miss Aulia. So I would like to thanks here still we have our speaker, Dr. Arnold Bovi. It was very interesting talk, and then many questions. I hope that it can be follow up by the collaboration with him in the near future. Okay, everyone. Assalamualaikum warahmatullahi wabarakatuh. Good evening, grace and peace, everyone. I'm Ahmad Fatoni, representative of Dr. Iman Hidayat, Chair of the Research Organization for Life Sciences and Environment BRIN. It has been a pleasure to be with you since yesterday in the 9th International Symposium of Innovative Bioproduction Indonesia on Biotechnology and Bioengineering 2022 with the theme of strengthening bioeconomy through applied biotechnology, bioengineering, and biodiversity. Thank you everyone again for attending this symposium. On behalf of the Research Organization for Life Sciences and Environment BRIN, I would like to express gratitude to all keynote speakers, invited speakers, presenters who have shared their idea, knowledge, and finding with us during the symposium. Since yesterday, I observed the excellent presentation from the great work of everyone and active discussion among our speakers and participants. So this symposium covering 6 topics, including bioresources and biodiversity, nutraceutical and natural products, biotechnology and bioinformatics, medical sciences, agricultural and forestry, and the last, environmental sustainability. So therefore, I can conclude that the purpose of our symposium this year has been accomplished. But however, I hope that this symposium is not just a tool for us to share our finding or idea and stop with the publication, but bear in mind that we still have a big responsibility to implement our findings to our any potential collaborators or stakeholders such as society, industries, and other users. Hence, our research hopefully can make a significant impact to the economy, society, and the nation or Indonesia. And one of the key success to achieve enough impact is collaboration. And I would like to take this word of collaboration as a key home message to all of us. Implementing our results and making an impact are not easy tasks, but I believe we are going to make it by working together with others. As our chairman of Research Organization for Life Sciences and Environment BRIN, Dr. Iman Hidayat mentioned yesterday in the opening session, that there are 3 factors that we should have done. Actually, first, working together with colleagues from other disciplines. The second, working closely with industry, and the last, mastering key technologies in order for us enable to utilize our biodiversity sustainably. So I do really hope that after this symposium, everyone, we would extend our collaboration with other colleagues that we just met during this 2-day symposium, or even with our keynote speakers from Indonesia or overseas. And here we still have Dr. Bovi. I hope that in the near future, or Dr. Wahyuni already have collaboration with Wageningen and others. Hopefully we can strengthen the collaboration with the Wageningen University. And I believe also that researchers and participants here already have their own collaboration, but I think there should always be a room to enhance our collaboration. Okay, before I close my talk, congratulations to Pak Sugiono, who have won the best presenter, and hope it will give motivation to us to get better and better in the future. Lastly, I would like to thank the entire committee of ISIBio 20. Oh, sorry, stop my video. Yeah. Can you see my video? Yeah, lastly, I would like to thank the entire committee of ISIBio 2022 that have done a fantastic job. They have spent their time, thoughts, and energy to prepare all of this. Thank you, Dr. Nani, Dr. Wahyuni, and all team members for making this symposium a successful one. Also, thank you to our collaborators, Indonesian Biotechnology Consortium, Consortium Biotechnology Indonesia, and other who have supported this event. Again, to all participants, thank you very much for your attending in ISIBio 2022. And finally, I would like to close my remarks and officially end the 9th International Symposium of Innovative Bioproduction Indonesia on Biotechnology and Bioengineering 2022. Wishing you all a success and hope to see you in the next ISIBio next year. Inshallah. Thank you. Assalamualaikum warahmatullahi wabarakatuh. Stay safe, everyone. Thank you.
Thank you
I
for
give
the
back to
remarks and also officially closing ISIBio 2022, Dr. Ahmad Fatoni. Ladies and gentlemen, that was the last agenda of these 2 days, ISIBio 2022, as well as mark the end of ISIBio itself. On behalf of official committee, I would like to sincere highest appreciations and also thank you so much for your attendance and active participations in all sessions during the conference held. See you on the next ISIBio next year. Stay safe and healthy.
Okay, thank you everyone. Yeah,
Thank you.
do all the best. And see you next year. Yep. Thank you.
Can you still hear me?